Recording. Morning, everyone. I'm going to step up here because unfortunately the bus is online and the mics are not working, even though they're green. So good morning, everyone. Welcome to the January 2026 TAC meeting. We're going to start with introductions. So myself, Kelly Hammer-Levy, I'm co-chair of the Technical Advisory Committee.
And Ryan Moyer, co-chair of the Technical Advisory Committee.
Mandy Deborah, I'm with Florida Fish and Wildlife Fisheries Monitor.
Jeremy Brown, Clearwater Deputy City Engineer.
Matthias Kramer, FWC Research Institute, specifically within the Coastal Wetlands Research.
Fish and Wildlife Research Institute, Coastal Wetlands Research. My name is Casey Craig.
Kara Radabon with FWC Coastal Wetlands.
Sheila Scar Moffitt, new name, with the Tampa Bay Estuary Program. Not all the presidents.
Jessica Lewis, Tampa Bay Estuary Program.
Stacey Dave, Kellis County Environmental Management.
Tracy Hurst, I'm with the Corps of Engineers in their Tampa permitting section.
Tom Reese, Ecosphere Restoration Institute.
Alyssa Powers, Nancy County Natural Resources.
Randy Runnels, Tampa Bay Park Reserves, FDEP. Ray Gribble, DSA.
Blake Simmons, Tampa Bay Estuary Program.
Michael Middlebrooks, Biology Department at the University of Tampa.
Scott Ditchy, Geosynth.
Hey, Lloyd, Citizen People Bird, PBS.
Brody Johansson, no affiliation.
Gary Lopez, FWC Hab Group. Sarah Kaminsky, FWC Hab Group.
Tiffany Lane, U.S. Fish and Wildlife Service Coastal Program.
Abby Kuhn, Oyster River Ecology.
Damon Moore, Oyster River Ecology.
Harry Thodiwali, Tampa Bay Estuary Program.
Mark Speck, Tampa Bay Estuary Program.
All right. Well, good morning, everyone. We also have a group of folks online. Welcome to the meeting today. Unfortunately, again, because of the microphone issue, the only voice that the folks online can hear right now are mine. So we're going to go ahead and get started with Dr. Carrie Lopez and her presentation. But when we get to the Q&A section, so if you guys ask a question, the speaker is going to have to repeat that question so that the audience can hear. So we'll try to remind folks of that. I know it's going to be a little challenging, but we're going to try to make this work. So Dr. Lopez, if you'd like to come up.
Okay.
Just wait a second. Yep.
Thank you. And although I like the title doctor, I don't actually have my PhD. I just I do like the sound of that though. I'm going to try to talk into the microphone so people online can hear me. Feel like I'm sort of leaning over though. So just, I guess if you can't hear online, just let someone know in the chat. Okay. So yes, today I'm going to be talking about short-term and seasonal drivers of Pyridinium bahamense bloom variability in Tampa Bay. And I'm going to focus on three questions in today's discussion. the first one is a bit of a review talking about what are the factors that drive pyridinium bloom phenology, so that natural cycle of blooms. And then looking into what are the factors that drive short-term variability, so variability within a bloom, and interannual variability of blooms in Tampa Bay. And then the last question is, why have we not seen pyridinium blooms for the last 2 years in Tampa Bay? And so again, a bit of a review, but just to get us all on the same page, what is pyridinium bahamense? It's an armored dinoflagellate. So in the first photo, we see 2 cells, and you can see that armor. It uses flagella for motility, so it can vertically migrate in the water column. It also has this thick-walled resting stage, which is the second photo, that spiky cell. And so that is how it overwinters in the sediment, and that is part of its life cycle. And then also just want to mention that pyridinium does produce saxitoxins, which can accumulate in shellfish and harm other wildlife and pose a potential public health risk. And just quickly, we talk about pyridinium being a warm, wet climate dinoflagellate. It has a subtropical to tropical distribution, and this is just to show that global distribution of pyridinium. And then when we look at the distribution in Florida, we can see that we have pyridinium occurring all along the coast. All these dots are observations of pyridinium over 10 years. But then we have certain areas that are hot spots, as you can see by the red and the orange and the yellow color. So Indian River Lagoon, Old Tampa Bay, Pine Island Sound, and also Pasco Zone and some areas in the Panhandle. And just to show that seasonality in Florida that we mentioned, this plot shows abundance of pyridinium on the y-axis in 4 different regions of Florida. IRL, Tampa Bay, Pine Island Sound, and the Pasco Zone from the years 2015 to 2024. And this is just to illustrate that we do see the same seasonality. And the red line indicates a high biomass bloom. So we have different magnitude of abundance in the different areas, but we have the same seasonality. And so I just want to talk a little bit about what drives that seasonality. And so I mentioned that pyridinium has a life cycle, and that is one of the drivers of this natural bloom cycle. So I'll just start with summer up on the your upper left, and that because that's when we see the bloom peak. We have the warmest temperatures in summer, so which support fast cellular growth rates, and then toward the end of the summer, the blooms typically end. And at that point, cells transition from vegetatively dividing to a sexual reproduction where they fuse and form the resting stage. And so then we have the resting stage in the fall and winter in the sediment, and that resting stage is dormant, and they can't germinate until they're exposed to winter temperatures. And those cool winter temperatures break dormancy so that in late winter, early spring, this can start to germinate again. So we usually get cysts germinating in February and March, and we start to see cells in the water column after that. And as temperatures start to warm in the spring in the water column, then we have faster cellular growth rates. And the process of germination is faster as the temperatures start to warm as well. So the take home from this is that the timing of blooms is pretty predictable, but the severity of the blooms is not. And so this is just a similar plot as I showed earlier, but just for Tampa Bay to show. So we have abundance on the y-axis for the years 2014 through 2025. And a couple important points to note is that the orange and the red are the high biomass blooms. So we have observed high biomass blooms in Tampa Bay almost every year since 2000 until 2024. So 2024 was the first year we hadn't observed a bloom since 2007. Then again in 2025, we didn't have a bloom. Another important point that is hard to tell from this figure, let's see if I have a pointer. But in 2023, in July, usually when the bloom is peaking, we had a sudden termination or crash of the bloom where cells went from over 1 million cells per liter to zero within a span of 2 weeks. And then they were absent from all of our water samples for several sampling periods. And then there was a slight resurgence, and that was really unusual. And this is the same data points, just shown differently so that you can see the spatial distribution. So this is with latitude on the y-axis. And so that dashed blue line represents the southern boundary of Old Tampa Bay. And you can just see that the blooms occur in Old Tampa Bay, although we do see some cells in lower and middle Tampa Bay. And just another point about this figure and the previous one is this is all the samples that we receive, not just our routine monitoring. So this would be samples from volunteers or from other agencies that samples get sent to the microscopy team. So you can see in 2021, we have a lot more sampling in lower Tampa Bay and middle Tampa Bay because that was following the Piney Point release. And then there was also a red tide. So there was more sampling in those areas that year. Okay. And so we wanted to look at factors contributing to bloom variability on short time scales. So the next few slides that I'm going to show are summarizing the results of an effort to investigate the environmental drivers of variability in blooms. And to do this, we looked at a routine monitoring station. So 13 years of data only from our fixed stations. And those are shown here in this map, 9 stations in Tampa Bay, indicated by the blue and the red circles. The red circles indicate our hotspot stations where we see blooms most often occur. And so what we did was we fit a series of logistic regression models looking at trying to estimate bloom probability from these basic environmental parameters that we monitored. And then we also included rainfall from the 3 airports shown. One side note is that you'll notice that we don't have hydrodynamics, residence time, factors like that in this model. We know those are really important and influencing bloom variability, but they're best addressed using other modeling approaches. And like I said, we wanted to focus on the relationships between these basic environmental parameters that influence the biology. Okay. Also, we looked at resting cysts in the sediment. And so the best fitting model showed a complex relationship. So this is like the terms that were in the best fitting model. So we had a complex relationship with temperature and salinity, a positive relationship with 14-day rainfall prior to the sampling, lag bloom, which was just indicating that the presence of a bloom the previous sampling day was important, and the cyst classification. So higher cyst, stations with higher cysts were more likely to have blooms. And so this is just, these 3D plots are a representation of that model output, showing the relationship between bloom probability as a function of temperature and salinity on these, I guess that's X and Y. These examples are showing as a function of, or scenarios of low rainfall, which would be a 10th percentile event, and high rainfall on the right, which would be a 90th percentile event, as a function of low cyst on the top and high cyst on the bottom. And you can see that, so with temperature and salinity, there were optimum values above which bloom probability would start to decline. And then the positive, you can see the positive influence of high rainfall events, so slightly higher chance or higher probability of blooms after high rainfall events. And then the effect of cyst was more dramatic in that we saw much more likelihood of a bloom happening if the stations were classified as high cyst. And we also used these model simulations to look at variation in bloom severity among years. And I've picked a couple of years just to show you. And so these are predictions of probability for the hotspot stations. That's what the panels are. For at the top is the 2018, which was a severe bloom year, and 2024, which is a non-bloom year. And so we can see that, oops, sorry. In 2018, we had very high probability of bloom predicted, and the circles at the top of the plot are showing that we did observe a bloom on those sampling dates. So that's just showing the relationship between observations and then our predictions. And then in 2024, a much lower probability of bloom occurrence, especially in the springtime. We did see an increase in probability later in the summer due to late summer rain events, but at that point there was not a bloom present, and we were past that point of when germination occurs, so there wasn't a source of cells. So we also wanted to look at how the environmental variables in our study were influencing annual bloom severity. So we created an annual bloom severity index that captured the magnitude and duration of the blooms. And those are shown here with the index on the y-axis over the course of our study period. And you can see we had a period of 5 years where we had above average severity. 2018, 2021 were our most severe bloom years. And so we looked at different factors like rainfall, salinity, temperature, and we also looked at nitrogen loading to see what the relationships were between that and bloom severity. And we looked on the annual scale and on the seasonal scale. And so the only relationship that really fell out for us in our analysis was a relationship between spring rainfall and spring nutrient loading. And so these plots show at the top, we have OTB specific nutrient loading. And on the bottom panel, we have OTB plus middle Tampa Bay and lower Tampa Bay. And as you can see, we didn't have a relationship with OTB specific nutrient loading, which was surprising, but we did with the Baywide. But I wanted to point out that there were a couple outliers that, so the most severe year was 2021, which had a lower than average rainfall in the spring, but that was also the year that we had the Piney Point release. So so the OTB specific nutrient loading was lower, but we did have higher than normal nutrient loading in lower Tampa Bay. But the relationship here with the spring rainfall and spring nutrient loading does make sense because this time period is critical for the development of the bloom. So it's important to have, the right temperature, salinity, nutrients, light, all those things that the Pyridinium needs to grow in the springtime when the bloom is developing. And when we looked at summer loading, we did not see a relationship either with OTB specific or Baywide nutrient loading. And we also did not see a relationship between rainfall. In our study, summer was June, July, and August. And again, I think if the important take home from this is that it's not just one thing and that the timing is important. So often the rains in the summer may come after the bloom has had the chance to develop. And so you don't necessarily see the response of Pyridinium because of the timing. So a summary of the environmental drivers from this study is that we have multiple lines of data. The annual cycle of temperature controls bloom phenology. And so with that background, we then can look at what factors influence bloom development. Salinity, temperature, rainfall, cyst abundance, and previous bloom presence are all important for bloom development and for bloom maintenance. So the conditions in the spring really set the stage for the bloom and may influence annual bloom severity. And the conditions in summer are important to maintain a bloom, but we need that precursor of cells. And so that brings me into the role of cyst as a source of cells and one reason we think that we have not seen a bloom in 2024 and 25. I mentioned 2023. So this is just a snip from the earlier plot showing Pyridinium abundance for the years 2022 through 2025. And I mentioned in 2023 that we had a unusual bloom crash where cells just completely disappeared and were gone for several weeks. So that suggests that they probably weren't able to enter their sexual reproduction phase because of that rapid crash. But they did resurge later. And so it is possible that there were, a reduced input of cysts that year. When we look at cyst maps, taken in 2015, which is the left panel, and compared to a cyst map in 2024, this is fall of 2024 though, we see that we do have a dramatic decline in to where almost all the stations are less than 500 cysts per gram of sediment compared to 2015 when we had over 1,000 cysts per gram of sediment at many stations in Old Tampa Bay. And when we look at the average cyst abundance over all the years we've collected, we did see a significant decline in 2024. And so what caused the decline, we're uncertain. It could have been lower cyst input from 2023 and then the no bloom in 2024. There could also be, cyst burial. And this is an example of where looking at the hydrodynamics and the interaction of hydrodynamics and biology would be beneficial. So just to kind of illustrate what we would expect for bloom probability with that observed decline in cyst abundance, I'm showing again the predictions for 2024 with high cysts in the top panel and low cysts in the bottom panel. So we're reclassifying based on that 2024 fall map, and we see a much reduced probability of a bloom in 2024. And then that coupled with less than optimal conditions in the spring likely decrease the competitive advantage that Pyridinium has in Tampa Bay. And so I think this is definitely part of the story, but probably not all of the story, because even though cysts are an order of magnitude lower at many stations in Old Tampa Bay, they're still not lower than we see in areas like Pine Island Sound and the Pasco zone. So the fact that we virtually saw no cells, we had one sample with cells in 2025, it seems like something else is likely also contributing. And again, I think, other modeling approaches that look at mechanistic factors would be really helpful for teasing apart some of the other factors contributing to this. So going forward, but like I said, cyst abundance in sediments are lower, but they're still there. And they're also still viable because we did germination assays in spring of this year. And so cysts are still germinating, are able to germinate. So the bloom potential exists. So we have to keep that in mind. And like I said, we need a better understanding of what caused the cyst decline and how ephemeral that is. If, one bloom will replenish the seed bed, we don't know. I suspect that it would build up over years, and that's what before over, since 2000. But the third point is that we need a better understanding of the complexity of the relationship between nutrient loading and bloom severity. As we all know, this is a complex question, and there's lots of interacting factors that make it difficult to tease out. And then the last point is just the bottom plot shows the chlorophyll index that we calculated in a similar way as the pyridinium index. And even though we didn't have a bloom in 2024, the chlorophyll index was not necessarily lower than average. It was just right at, average, really. And that was because we had blooms of other dinoflagellates. And so that's just something to consider going forward. That's all. I'll take any questions.
Okay, with the cyst and sediments, you had a point up there about sediment burial. What is the activity of cysts in the sediments? Are they traveling from superficial sediments deeper? What's the mechanisms for activity and transport with the sediments?
So we know from previous.
Can you summarize the?
Okay.
People here on actually.
Oh, sorry.
Awesome.
Okay.
Could folks online let us know if you heard Brandon's question?
I'll just briefly summarize. The question was about.
We can hear.
Okay, good. So we know from previous work that there is in the upper, where we focused on the upper 5 centimeters, there is exchange in that zone, mixing through resuspension events or just a biological mixing. We didn't look at the mechanisms, but we worked with USGS and they did short-term radioisotopes. So we know there's like mixing in those top layers. But so there, and the cysts, we have cores that show the cysts go deep into the sediments. But the cysts need oxygen to germinate. So the very upper layers of the sediment are probably what are available for germination because that is the trigger. So once they're not dormant and they're able to germinate, the oxygen is the trigger for them to begin that process. And one thing I didn't mention is that we did get two cores this year that we're processing now with Eckerd, and we want to compare those cores with cores we took years ago just to see what's happening in the recent sediments. And because the sediment, the cyst samples I showed were from grab samples, which are, scooping, like combining several, like 5 centimeters or so of the top layers of sediment. So we're trying to get a closer look at the small intervals in the top layers.
Thank you.
If we have warmer water temperatures during the winter, would that be better for the blooms? Like, do you have to have a certain low water temperature to then promote the?
That's a really good question. So in subtropical zones, or at least in Florida, it seems that the cool exposure in winter is necessary for cysts to break dormancy. But pyridinium also blooms in tropical regions, but in those regions, it's typically not as seasonal and it's more like cells would be more present year round. You may still get seasonal occurrence, but I don't know if the triggers for germination are different in tropical zones or if the vegetative cells in the water column play a more important role in seeding those blooms in tropical zones. But from our work of cysts in Tampa Bay, we know that they need a cool exposure to break dormancy.
Got so go ahead, Brandon.
One kind of general question on cyst Baywide. Do you feel that there's a a tipping point one way or the other where if you have a certain level of cyst present, you're going to maintain that potential for bloom, or vice versa. If you achieve a level, and how you would measure or verify that, of the absence of cyst, where you may reach a tipping point of reducing that bloom long term.
I think that would be a good question to answer with a model. I think what we can do is look at the data we have and sort of make assumptions based on where we see blooms and what the concentrations are there. And that's where I came up with the 500 cysts per gram cutoff for the high and the low classification. But like I said, in other, like in Pine Island Sound, the cyst concentrations we've measured are lower than that. So, and there are still high biomass blooms there sometimes. So there's other, the hydrodynamics, other factors that really you need a model, I think, to get a better answer, better hold on that.
Questions online. I think Frank, you were first if you want to unmute and ask your question. Otherwise, I can just read it out.
Yeah, hello, everybody. Can you hear me?
Yeah.
Well, great. Thanks. Beautiful work. Thanks for presenting it. You mentioned Piney Point, but there's many other things going on in Tampa Bay, including the many changes over the years up to the Howard Franklin Bridge. And I've seen, just driving over the bridge, some of the high sediment that occurred when they were moving sand and putting pylons in and so on. And then we've also had some hurricanes. I was wondering how you can bring some of these other factors to have a more comprehensive picture of what may be controlling the blooms.
That's a really good question. And I think it's easy to miss these types of events and analysis like this because some things you might not even know about or have a good record of, like sediment transport and things like that. But I've wondered that as well, and like what affect bridge construction. There's just, I think it's really difficult to capture all of the factors contributing, but I think looking at outliers is one way to begin to understand how these different sort of sporadic events are affecting blooms, because we're looking at data that we have regularly collected, but then we get these outlier points. And I gave the example of 2021, but I think that may help us explain things like that. Looking at those outliers may help us understand the relationship better and why we have such difficulty capturing the relationship. I didn't really answer your question, but I think my answer is that's a really good and overwhelming thing to consider. Yeah.
I'll say too, in regards to the hurricanes, I mean, you had the plot of chlorophyll in 2024 versus the bloom severity, and the hurricanes came in the end of the year after typical bloom periods of pyridinium. So when we saw the chlorophyll spikes in 2024, it was late in the year.
Yes.
Yes. An interesting note about that.
Right. As well. Right. Yeah, that's a really good point. And I think often when we were in years where there were pyridinium blooms and we were looking at the relationship with summer rains, that also was a factor in that those rains came later after maybe the bloom had its chance. So in that example that you give, there were other dinoflagellates that bloomed right. But even in years with pyridinium, the timing during summer, I think is, important. So maybe looking at a 3-month, period is informative, but it's not telling the whole story.
There's one more question online. CG Smith, if you want to unmute and ask. I could paraphrase. There's two questions. Did you have any wind or wave patterns with the logistic models? That's the first one. I'll stop there.
Yeah, no, with the logistic regression, we focused on the environmental data that we had in the time series and like the rainfall that we could get along time series. And we did not include hydrodynamics, but as I mentioned in the presentation, that is an important thing to look at. And while I didn't think it was necessarily feasible to incorporate in the logistic regression because of the data availability, that may be wrong. There may be a way to incorporate it. But I also think looking at that with mechanistic models is a good approach.
All right, second question. Along the coast of 2023, salinity was very high in June and early July drought year. Would that also affect cyst production?
I'm not sure about cyst production, but I think it did affect the 2023 bloom. That bloom started off early and strong, but like I said, it crashed. And I think that was likely due to that it was such a dry year. That was the question?
So,
yeah. And so as far as cyst production, I think the abnormal termination of the bloom and the inability for them to necessarily transition into their normal sexual reproduction, which is speculation. We don't have evidence of that, but just because of the way it ended or crashed so quickly, I think that could have influenced this production.
Thank you.
Thank you.
Yeah. So if the room mic was working here, our next presentation is by Dr. Michael Middlebrook. He's going to tell us who or what has been speaking for our today.
Okay, so I don't need to. All right. Deal. I'm not going to stoop over. Hello, everybody. Thank you for your time. I didn't put any pictures on the first slide. I don't know what's coming. It's more fun for this. I'm Michael Middlebrooks. I'm a biology professor at the University of Tampa. So my primary job is teaching invertebrate zoology courses there. But I also do some research, of course, and I'll tell you a little bit about some of the research that I've done along with John Ambrosio and Susan Bell. So my research focus, the thing that I'm most well known for and what I do most of the time is the sea slugs. So I work on natural history and ecology of mostly Stachyglossus and sea slugs and nudibranchs as well. They're very interesting organisms, but I'm also very interested in seagrass ecology and restoration. And so that's what I'm here to talk to you all about today is some seagrass project funded by the Tampa Bay Estuary Restoration Fund. So just to dive into this a little bit, some background. Generally, we want to have lots of seagrass in our shallow water ecosystems, right? For a variety of ecosystem services and natural healthy habitats. A lot of the areas that I'm looking in and focusing on today is looking at Paluduli wrightii, the shoal grass beds in particular. There's a lot of things that can happen to cause seagrass loss. So it can be a number of different disturbance events, eutrophication, whatever that is. And when the seagrass dies back or is lost for whatever reason, a number of things can happen, right? We can have bare sediment, but in some instances, we have macroalgae growing in this place. So what we will see sometimes in Tampa Bay is when we lose Paluduli wrightii for whatever reason, it can be replaced by this algae Caulerpa prolifera. And that does kind of look like a seagrass there, but Caulerpa is not a seagrass. There's some similarities between the two in sort of the overall structure, but it is a little bit, it is different. It is a green algae, so it doesn't have the vascular tissue that you would find in a true plant like seagrass. Like seagrass, most of its growth is going to be vegetative. It does have a sexual reproductive cycle, which is quite complicated, but mostly it reproduces asexually just by vegetative growth, will colonize new areas by fragments breaking off and growing in new places. It's naturally part of our ecosystem. So it is there as a, usually as a smaller understory as part of this in the seagrass beds. It's not an invasive species. So there are some caulerpas that can outcompete seagrass in some areas where they're invasive. Caulerpa taxifolia in the Mediterranean is quite famous for that. But this is a native species. This is a normal part of what we would have in Tampa Bay. You'll also notice that the below ground structures are quite different on this. So it doesn't have the complicated root system that you would see in seagrass. So you won't get quite as much sediment stabilization, might have different invertebrates living in the in fauna or the under the sediment there because of that difference in structure. But the blades that it makes can provide a nice habitat for a lot of organisms. So this could be potentially a nice alternative habitat for areas that have lost seagrass and for a lot of those organisms to live in. So it can provide some of the same ecosystem services that you might want from seagrass, and can provide habitats for a lot of those little organisms that live in there as well. One other big difference about caulerpa is that it is a cenocytic algae. So this is not a picture of caulerpa. This is a picture of Bryopsis, a different species of algae, but caulerpa is a bit too thick to see this under the microscope. What we're seeing here is an algae that doesn't have separation between its cells. So you can either think of these things as one giant cell with lots of nuclei inside of it, or lots of cells that just don't have the separation between them. So it's kind of a weird thing, but this is in biology, there's a bunch of instances of this in all kinds of different organisms. Some of the green algae do this as well, and caulerpa is one of those. That is important to keep in mind for a little bit later, what I'm going to be talking about. But basically, there's no individual cells here. So there's been a lot of documented caulerpa prolifera blooms in Tampa Bay. This is data that originated from swift mud surveys here. From just some different site locations that I think from the past 25 years or so, where this has been documented. This isn't necessarily all of them, but these are just an example to show you some of the locations where we've had some blooms of caulerpa in those areas. So, and this is the site that I'm specifically going to be looking at here in Old Tampa Bay. So we'll be talking about some of the surveys that we've done in this particular area, just to kind of give you an idea of where we're looking. So the goals of our study were to compare the macroinvertebrate communities between Haliduli wrightii and Caulerpa prolifera. So the site that we picked was previously the area in it dominated by Haliduli, the shoalgrass, and then some of it died back for what reason, and then Caulerpa grew there. So when I was looking for sites, I found an area that had lots of Caulerpa in it. In the beginning, there was Haliduli there, but it was a bit sparse. And so we want to know how similar are the communities created by seagrass and algae. How useful is Caulerpa actually as a habitat for these small macroinvertebrates, all the tiny little critters that live in the seagrass and mostly get ignored but are quite important food web. And then we were also trying to conduct some recolonization experiments in a mixed Haliduli wrightii and Caulerpa prolifera bed to see how, basically make some little patches and see who grows in there first, right? That was generally the idea. And then the Caulerpa disappeared in the middle of this study. So we did manage to collect samples to get some of the macroinvertebrate data, and we're still processing that, and that's actually not what I'm going to end up presenting today, because I'm going to tell you about what happened. This is sort of a serendipitous event. So we managed to catch this happening, the Caulerpa dying off. So when we went out, we started this study in January of 2023. These are just some means, some averages of the number of Caulerpa blades from some core samples that we took. You can see in January, there's quite a lot of them. And then in June, there are that number has declined quite a lot. And this is maybe even a bit misleading, because these are targeted core samples where we're specifically going after patches of Caulerpa, and the Caulerpa patches were much harder to find in June. They were basically everywhere in January, and 6 months later, they were gone. not totally gone, but were declining. So I wanted to figure out what was happening, put on the mask and snorkel, swam around, and found some interesting observations. But this is what this looked like by December. We'll get to what we found here in a moment, but December, this is all Haliduli. I think there might be a little bit of Serengodium in there as well, but the site has changed quite a lot. So just some of the regrowth areas that we were looking at, right? We've got a fair bit, especially for December, of Haliduli in those areas now, when these were denuded in June. So they, had grown back, and there's no caulerpa at all at this point. I show you some pictures of it here, but there wasn't any to capture. And this has happened before, though. All of these sites that I showed you all previously where we had those caulerpa blooms, the caulerpa disappeared at some point. Since the 1999, there's been at least 24 instances of blooms in these beds. And then they disappeared at some point again. And how long they persist is variable. So sometimes it's 1 year, sometimes it's 7 years. This is probably happening in a lot of other places that aren't actually monitored as well. Just a quick example of two sites, sort of what this could look like over time. Again, this is data derived from some Swift mud surveys. Frequency of occurrence here, right, on the axis there, and what we're seeing, right, is you've got a couple peaks where we have a lot of caulerpa, and then it just drops off quite a bit. Here's another site, same sort of idea. This one's a little bit more variable. It's and these booms and busts of the caulerpa in that area. So we don't know why this happens, but what we found while we were out there was interesting. So what happened to the caulerpa? Well, it's sea slugs, right? And just so happens this is what I researched. This is we're out there finding a lot of little sea slugs eating the caulerpa. So they were all over it. There's sea slugs all over the site. And so we decided to take advantage of this, capture this moment, do some surveys, document what was left of the caulerpa, document how many sea slugs are actually in the area, and document the reproduction of the sea slugs as well. These type of animals that we're looking at here are a group called sacoglossans. So they aren't nudibranchs. If you've heard of nudibranchs before, they're a separate group of sea slugs that independently evolve the loss of the shell. They are quite a bit less studied than nudibranchs. Not that nudibranchs are particularly well studied, but these are a little bit less known animals, but they happen to be my area of expertise. And sacoglossans are specialist herbivores for the most part. So they feed on algae, and they're very picky about what algae that they will eat. For some of them, they will only eat one species of algae. For others, it would be just a few species of closely related algae. But it tends to be a very tight ecological relationship between the algae that they eat and what they are and the sea slug. And the way they feed is a bit unique as well. So they feed sectorially using a specialized radula. So the radula is the feeding apparatus in gastropod snails and slugs. This is an electron micrograph of Elysia chlorotica here. So that's what the radula looks like. And they just use one of those teeth on there at a time and basically poke a hole in the algae and then suck out the cellular contents. So it's kind of a very specialized feeding mechanism. I'm going to show you what that looks like in one example. This is not the animal we were looking at, but this is one I have a decent demo of. This is a little tiny one called Styliger. It feeds on this red algae called Ceramium. And here's a little video of the slug. It's adorable, right? It's going to poke a little hole, shake that algae because it's sucking out the contents there, right? Very cute little guy. Eating. I filmed that under the microscope. So it's quite a small animal. And then after it's fed, you can see the ceramium, right? All the cellular contents are gone. Now, that's algae that has separate cells. If your algae does not have separate cells, if it's acidic like the caulerpa we talked about, you can just poke a hole in any spot on that and basically suck out the whole thing like a Capri Sun, right? They're able to feed on all of it from one spot. And that's what we're seeing here. So the animal that we found is one called Elysia subordinata. It is one of our larger Sacoglossum species in the area. So it can get to a fairly decent size, although it will sort of depend on how old it is and how much they've been eating. They're a beautiful, interesting slug, and they are a Caulerpa specialist. So they will eat multiple species of Caulerpa. I think about 9 have been documented from feeding on them, including Caulerpa prolifera. The image there has them on a different species of Caulerpa. But they are Caulerpa specialists. So that is the only thing they can eat. They can't eat seagrass. They can't eat other algae. It's only Caulerpa. That's the only thing that they will recognize as food. And so we went out and we did some surveys. So we estimated the cover of Haliduli, the seagrass, and Caulerpa, the algae out there using quadrats across 4 haphazardly placed 40-meter transects. So 80 total observations here. Counted the number of Elysia subordinata, the sea slug, in each quadrat, as well as the number of their eggs that we found. And this is what we found, right? 78 of the quadrats contained Haliduli at this point. So with a mean cover about 27%. Only 22 of them contained Caulerpa with about mean cover about under 5%. So big reduction in our Caulerpa cover. And where we found Caulerpa, we found lost slugs. 194 individual slugs were found, and 113 of their egg masses were observed during the survey, including some instances where we saw them depositing the eggs while they're out there. Both Caulerpa and the slugs have been completely gone from the site since December of 2023. I've been back every 6 months. Last time I was there was in this June of 2025, and no Caulerpa, no slugs. So this pattern has continued for at least the past 2 years. Just some images from the surveys here. So a lot of the slugs were on bare sediment. We found some of them crawling on the seagrass as well, which is what the 2 photos on the left are showing you. But they are not feeding on it. It's not food for them. They can't survive there. Realistically, these slugs consumed all of the algae, and then that's it for them. They will starve after that point once all the algae is gone. But they put a lot of that energy from the algae into reproductive output. So this is some slugs engaged in some amorous behavior. They are simultaneous hermaphrodites. So as long as they find one other individual of the same species, they can mate. And there were lots of individuals. So there was lots of mating and lots of eggs. So this is a photo of what they look like when they're laying eggs. This is one I took from another study in an aquarium years prior. So this is not one from the field. But here are some eggs from the field. So this is one that I found on just a leaf that came from the land that was in the water. Here's one laid on some of the seagrass, right? So a bit different shape than we would typically see, but they're laying lots and lots of eggs. So not a small amount of reproductive output here. So where does this leave us? It's kind of an interesting observation, but so the slugs have completely removed the Caulerpa from the study site, and neither have returned as of June 2025. The question, I think what's really interesting here is, could herbivory from these sea slugs explain the other instances of Caulerpa loss that we've seen in Tampa Bay? I think that's something that we should start looking for. It is a bit difficult to spot sea slugs when you haven't learned how to do it yet. They can be a bit cryptic. They will look like the algae that they're on often. But if we can start documenting when we find them, if we find them in conjunction with the Caulerpa, and if large numbers of them, we might be able to better predict when we'll see these busts in the cycles of Caulerpa. There's a report from 1990 in the Indian River Lagoon that found a lot of Elysia slugs. It was an unidentified, they didn't identify the species. I suspect it was Elysia subordinata. There is one other candidate species as well that it could be, that might have been responsible for a big loss of Caulerpa prolifera there as well. So this may have happened in other locations too. There's a couple other wrinkles to this though. If you read the scientific literature on Elysia subordinata, the reproductive biology for the animal is listed as being lacithotrophic. And basically what that means is they produce big eggs with lots of yolk in them. And so the offspring get lots of energy from their parent, right? And that translates into either a very short larval stage, or in the case of this animal, the larval stage happens complete within the egg capsule, and so they just hatch as a juvenile slug. So very low dispersal from this reproductive method, but it is you make bigger offspring, so potentially higher survival if there's food for them in the area. Now, what we saw, there would not be any food left for those offspring. But coincidentally, I happen to have been working on a study on some of the reproductive output in sea slugs, and we've learned that when I collect these animals from the Florida Keys, their developmental pattern follows what's in the scientific literature. When I collect them from Tampa Bay, they have what are called planktotrophic larvae. So they make a lot more eggs, but the larvae hatch as a swimming veliger larva. So it is actually dispersing. So even though the adults completely wiped out this caulerpa from this spot, their offspring have the potential to find a new location because they have dispersing larval stage. So that population is not doomed from the gluttony of their parents. They could actually find a new location and survive. And this is something that I think gets missed a lot for a lot of invertebrate animals. Somebody does the work once, you assume it's the same across their entire range, and it may not be. And so this is an example of just that. We do also have 2 other caulerpa eating sacoglossans in Tampa Bay. There is one other Alicia species that's been documented in Florida that could be eating the caulerpa as well, but I've never seen them on the West Coast, so I don't know if we have them. But we do have these 2 here. One Lobiger Seruii is a very beautiful slug. I've only ever seen them on Caulerpa racemosa, so I don't think they're affecting this particular species of caulerpa, but they might. However, Octonoi antillarum does occur on Caulerpa prolifera, and they will feed on it. I've maintained them on it in the laboratory. They're quite happy to eat it. They'll reproduce on it, and they also have a planktotrophic larva. So this is another one that could be doing the same kind of thing. So it may not always be the same species. We might get other herbivores doing that. And I think I forgot to mention this before, but caulerpa is in general a bad food for herbivores. It's highly protected from defensive secondary metabolites, so chemicals that make it taste very bad or make it toxic for organisms to eat. These sea slugs are specialized in it though, so they can handle the toxins, maybe even incorporate them into their own biology and use that as a means of that. So you're not going to see a lot of other herbivores consuming the caulerpa. Really, these guys that are likely to be doing that. So with that said, I think this is an interesting thing. We need to learn more about it, and I'm hoping that with some more people keeping an eye out for the slugs, we can continue to see, is this an isolated incident, or is this what drives this pattern that we see over time? Some acknowledgement. So some of my collaborators here, Susan Bell, John Ambrosio, and Hood. I had a lot of students working on various aspects of the project from the University of Tampa. So thank you to everybody and all of my students, and thank you all for your time. Take any questions if you have them.
It's since not many things eat the prolifera, have you ever noticed them laying eggs on prolifera blades, since it's not likely to be disturbed as much as
Yes, I definitely in less dense populations of slugs, it's very common to see them lay eggs on the algae that they feed on. So I've seen that on a number of different caulerpa species, but not in situations where we've had such a high density of slugs. I think they probably would have been doing that if there was more caulerpa left.
Could you plant the sea slug so that you could change it?
I would. The question is did I plant the sea slug so I could change projects? And the answer is no, I'm not that clever. I really was trying to take a break from doing some sea slug research, and they won't leave me alone. So,
I don't know if I forwarded it to you, but there was a manatee something related meeting where he documented something really similar happening in the Indian River Lagoon, I believe it was. So I'll send that to you if I can find it.
Yeah, that'd be great.
Yeah, it's
definitely interested. I think the ecological role of these animals has been underlooked, and it may not be. It may be a rare instance, or this might be really what's driving it. These animals are really easy to overlook. They're not very big. A large one is maybe a few inches, and most of them are going to be smaller than that. So, question, yeah.
I'm curious from like a management perspective, would it be possible to culture these organisms, not necessarily for prolifera, but for fastigiata? Like that one is like, I know really bad in Charlotte Harbor too, to like mat over huge areas of seagrass and cause a huge issue with that. And I've like seen other areas where I've seen fastigiata come over, mat over things, and then I'll see it was the one on the right of the second one, the oxyno or something like that. Where those things that is what really does seem to be kind of from like an observational aspect, what drives the die-off. So like when you show like the little cysts of the other species, like would that be something from like an aquaculture perspective you could culture that quickly so that if you see a bay-wide bloom, the east wall of Charlotte Harbor or something, you could go out with sprinkle these little guys out there and give them a jump start. It'll work. You'll be a millionaire.
The culture would not be easy to do. It is possible. So if the animals are making planktotrophic larvae, you also need to culture, microalgae for them to feed on in their larval stage. And those algae, the dealing with larval slugs is they're finicky. So it is possible to do it, but you would need a fairly robust setup to do that. I would be less concerned doing it somewhere with a native habitat. There have been some people suggesting to try this in the Mediterranean to try to clean up some of the invasive algae, and I'm always very cautious of introducing new species to areas. So I would be opposed to that sort of thing. But if within a native area, I think trial would be pretty cool study, worth to see if it's
like with Fastigiata, there's been a lot of like day wide, throw your hands up. What do we do? Wait.
Yeah. I mean, and so that might be a way to do that. It is technically possible. The ramping that up and keeping those cultures going would be a challenge, but it would be doable with the right facilities and personnel.
I don't know the growth structure of Caulerpa to say, but I assume they're feeding on the blades, and that's the cellular structure that they're sucking out. Does that extend to the subterranean?
Yeah, it's all connected.
All connected. So they're wiping out the whole thing, start to finish.
Yeah. So if there's enough of them, they can wipe out an entire bed pretty rapidly, and from
not just the cover that they're clearing out.
Yeah, it's all that below ground mass is all connected in these rhizophytic algae, and so they can that for the algae that can be good. So if an area gets wounded, they can devote some resources, sort of push it that way, plug it up and regrow. But if it's continual, that it's going to get wiped out from that standpoint.
One other question, kind of, and maybe the slug side of it took over, but back to the original aspect of what you were looking at with Caulerpa versus seagrass. You mentioned obviously there's some overlap in ecosystem services. Did you get far enough into that on any comparisons of Caulerpa beds versus Haladuli or other seagrass beds in Tampa Bay? And what is your thoughts on services provided by those two?
So I have some initial data on comparing some of the crustaceans and some of the gastropod communities between the two, and they're very similar. So there are some differences in the number of for a few species of the crustaceans. So there would be some that were more, I'm not remembering exact species on the moment, but some that were a little more populous in the Caulerpa, some a little more populous in the Haladuli, but the community overlap is more or less identical. So the differences that you would really see between the two are in specialist feeders so far is what we've seen. And so there is also a tiny Sacoglossum that feeds on seagrass, but they're very rare, and because seagrass does have individual cells, they're very small. So they're not really putting a dent in our seagrass. But that's one that shows up, for example, that's only in the Haladuli, would not.
And the site that you showed was it had gone from seagrass, which died off, Clerpa took over, that got eaten apparently, and then the Haladuli came back.
Yeah.
You know, I think there's two sides of that, whether macroalgae and Clerpa are areas that can stabilize sediments and systems, which can promote seagrass coming back if it's not outcompeted, versus taking over seagrasses where they've been established. So you feel like the elimination of the seagrass is mandatory in establishing that Clerpa in Tampa Bay specifically.
I think that's what the patterns are showing is that, yeah, we don't see the Clerpa. It's there. It's normal to see it as part of the system, but it's not really becoming a dominant ground cover until the seagrass dies back. Now, once it's in there, it might be preventing it might be impeding the space from seagrass really taking the area. I don't know that. And these ecosystems are complicated, right? We're not just looking at the interaction between those two species, right? It's all of the environmental conditions and variations that we're seeing. So, the nutrient input is going to have a big impact. Clerpa does well in higher nutrients than seagrass. It likes a little bit higher nutrients than seagrass often does. So that might be a factor as well. And there may be some differences in temperature tolerance and things like that too. So especially for these very shallow habitats like the shoal grass likes. Thank you. We had a question all the way in the back.
We're doing our studies in the Hillsborough Bay with the city of Tampa. We found many occasions when larvae came in really rapid, colonized the shallow areas, and then equally rapid disappeared. And we noticed Alicia a few times on those larval beds, but we think come up with a conclusion that major rainfall event wiped out the larva.
Yeah, and that may be as well. I mean, it's hard to pin any kind of big ecological event on a single factor. So it might be multifactorial things involved.
I think some of those studies are published in the basis for feeding.
Oh, great.
Like the rapid drop in salinity is what caused it.
Yeah.
I don't know. Do you know if manatees eat Clerpa or because of toxins they won't touch it?
So I've been reading about this, and in some studies looking at manatees and green sea turtles, when areas where they've lost a lot of seagrass and they had to switch to an algae-dominated diet, Clerpa still makes up a very small percentage of what they eat. So they will eat some, but they tend to avoid it. Probably because of the toxicity. Those large animals can eat a little bit and not, and they won't. Poison is all about the dose. So if it's a small amount, it probably won't harm them much. But if that was all they're eating, it would probably make them dead. And it may also just taste bad as well. So. And if you like sea slugs, I've got I have Instagram account and Blue Sky stuff with more sea slugs. I also have a TED Talk, which is about how some of these sea slugs are photosynthetic. So they actually make their own energy, not so much Elysia subordinata, but some of the other ones. If you're interested in that, there's more. Thank you all very much.
All right, 2025 State of the Bay.
In here.
Not going to be as fun as slugs.
Oh, come on. The State of the Bay is good. Okay, good morning, everybody. This time of year, I gather up all of the data products, graphs, tables that I work on every year and try to put together in a cohesive narrative about how the Bay is doing. So this is a kind of a challenging exercise because the pieces don't always tell the same line of information, but we try our best sort of give a sense of how well the bay is doing. And so this is a reflection on all of the pieces that make up the data products for 2025 in the context of long-term recovery and status and trends of the bay. So that's what this is about. We always start with the water quality report card. I printed a bunch yesterday. They're over by the door there if you haven't picked it up. But just to remind everybody, there's two sides to this report card. There's the management side, which is looking at kind of a report card that combines chlorophyll and light attenuation and reports it relative to how it may affect seagrass growth. And then the other side is a regulatory component where we look at chlorophyll as a function of whether or not it's meet the regulatory threshold for the different bay segments. And that's something that we communicate to the nitrogen management consortium at our last meeting in December. So on the management side, you're probably aware we have these three colors, again, as a reflection of chlorophyll and light attenuation. Green being stay the course, continue what you're doing, continue monitoring progress. Yellow being caution, look at the monitoring data, look at the nitrogen loading estimates, talk to the TAC and the management board, and the red being you have to really take note that something really is right here. So those are the three colors. They're not just good, okay, bad. They actually have management implications associated with them. But the good news is for 2025, looking at all of the four major bay segments, we are in the green. We have been for a number of years. I think actually last year, Lower Tampa Bay was in the yellow for a brief second, but for 2025, we are in the green for both chlorophyll and light penetration. We are below our management targets for both of those things. Yes, and so looking at it long term, yeah, so this is sort of the long-term history of the four major bay segments from 1975 to present, 2025, all the way down here at the bottom, all in green. So that's all good news. As I said, last year we had Lower Tampa Bay in yellow, which I think was kind of a function of the hurricanes coming through. I'll show actually I'll show a time series of the chlorophyll trends for each of the bay segments. You can see 2024, we were flirting right at the threshold for chlorophyll, but good news is 2025, we're looking good. As far as the regulatory standards, the thresholds are a little bit higher than the targets. So it's basically for that report card, it's whether it's above or below the threshold, just looking at chlorophyll A. Also, we're in the green for 4 consecutive years for the 4 major bay segments. So that's definitely good news as well from the management, or rather the regulatory standard. And then looking at the trends of chlorophyll. So these are the 4 major bay segments over time, annual average chlorophyll. We have Old Tampa Bay, Hillsborough, Middle, and Lower Tampa Bay. The lines are basically the management targets as the solid line there. And then the top dotted line is going to be the regulatory threshold. So obviously we're seeing here a picture of long-term recovery and water quality of the bay, but looking at just the last year alone, we are well below the management target and regulatory threshold. As I said, 2024 was an odd year because we had those hurricanes come through late in the season. That actually did bump up some of the chlorophyll concentrations above the management target. So the good news is that happened again late in the year. We were talking about that earlier, where we were kind of past the growing season. So it didn't really have a huge notable impact on water quality. It was just kind of that October, November, December period. But we did see an overall increase in the concentration. But because our report card does look at both chlorophyll and light attenuation, this increase in chlorophyll in 2024, you have to take that in perspective of light attenuation as well. But story for today, 2025, chlorophyll concentrations, at least on an annual perspective, are well below the target's thresholds. This is looking at it spatially. These are all of the EPC monitoring stations that we use to develop this report card. We don't assess individual station trends, if you will, or compliance if you're looking at their regulatory threshold. But this just gives you a sense of if we did have hot spots of chlorophyll, where they occurred, and for the most part, not really a whole lot going on relative to the targets. There was, this is a typical location where we do see generally high chlorophyll, but for the most part, not looking at the base segment average pushed out above the target threshold. And one little spot down here in the northeast of Tampa Bay. We also like to look at seasonally how the chlorophyll has come about. This is another plot I always show, but we have really for the four major base segments, the box plots are going to be the long-term monthly distributions of chlorophyll, and then the red dot's going to be 2025 relative to that long-term distribution. So when we do have exceedances of chlorophyll in Old Tampa Bay, typically, or when that does happen, it does result from high values in the summer that push that annual average above the threshold or the target. But this summer, pretty quiet. October, we see a slight little bump, but for the most part, all of the months are generally below the long-term seasonal values, median, if you will. So like these lines here are going to be the median from the long-term historical distributions from 1975. I talked about light attenuation as well. This is the other side of the coin when you look at targets in relation to how it may affect seagrass growth or a light environment for seagrass growth. So we also look at light attenuation trends, so how quickly light would attenuate into the water column. Not really a whole lot going on here, other than I had for years been talking about this kind of uptick in Old Tampa Bay, kind of showing a slight trend, actually exceeding the target, but it seems to have sort of reversed in recent years. Lower Tampa Bay is also kind of interesting. There's a slight trend approaching the target or threshold that we're keeping an eye on, but at least in 2025, none of these were sufficient to push it above target. What's also interesting too is, this is part of the narrative for this year is chlorophyll was generally across the board pretty low. But if we look at light attenuation, even though we didn't really, at least on an average for each base segment, exceed the target, there are a couple instances where individual stations showed up as exceeding that target, which is kind of interesting because typically we see, when you have high chlorophyll, the light attenuation is also high, but now we're kind of in a situation where we have low chlorophyll for 2025, but somewhat high light attenuation at some stations. So there is kind of this disconnect between the two that we're thinking is maybe driven by other factors, whether it's turbidity or color in the water column, not just chlorophyll. So we did look into that a little bit, but just to point out that there were a couple points in Old Tampa Bay where we did see high light attenuation. Also a couple stations in the middle. This is it seasonally. Again, not really much going on relative to the long-term trends. Light attenuation overall, pretty much these values have been lower than historical distributions. Okay, so why did we see good water quality in 2025? It's kind of been the story that we've been telling the last couple of years as well. There was some debate in the office about whether or not 2025 was a dry year, and it turns out it actually was compared to the long-term average. If you look at 2023, that was the driest year on record for, I think, the last 10 to 15 or 20 years. I forget the actual number, but very, very dry. Good water quality in 2023 as well. 2024 was kind of interesting. I think a dry year overall as well, with the exception of the storms coming through at the end of the year. That's why you see that average being above the long-term average here. But again, that happened late in the season, right? So that did contribute to a large nutrient influx into the bay from stormwater. So that's why we didn't see, extremely high chlorophyll. But 2025, dry year, chlorophyll was quite good. And so that's kind of, we think, the primary driver of water quality last year. So what does this mean for seagrass? So our whole management paradigm, right, is control external nutrient loads, reduce algal blooms in the water column through the light environment. See, that's covered. That's sort of the mantra that we've been using for a program for many years now. So when we talk about water quality, we also got to talk about seagrass. So as you might expect, because water quality has been quite good overall, at least from the transect surveys we collect every year, looks like seagrasses are trending in a positive direction. Whether or not this translates to a large increase in the aerial cover is another question. We have map 24 in the water management district where bay-wide there was an increase, but not super sizable. Old Tampa Bay is still an area of concern, at least from those maps. The cover still showed a decline, but just looking at the transect data, which is an alternative line of evidence here, bay-wide, it does look like we are trending in a positive direction, primarily from increases in algae, which, as we learned from the last talk, is coming back in some areas where it was denuded or lost. By bay segment, we have Old Tampa Bay, Hillsborough, Middle, and Lower Tampa Bay, and actually this includes Boca Ciega Bay as well. Old Tampa Bay, just looking at that, that's kind of an interesting one because we did see this notable drop. This is again still the transect data from 2019 to 2021, just looking at frequency occurrence. It does look like it is trending in a positive direction, similar to Bay as a whole. And again, this is likely due to the recovery of Haladuli. Hillsborough Bay is also interesting. We talked about this last year as well. This is where you look at the last map from the water management district. There's a huge increase in cover in Hillsborough Bay, although historically this part of the bay has not had a lot of seagrass, so overall it's not a huge gain, but for that bay segment it's a big deal. Basically there's just a huge increase in Haladuli at Hillsborough Bay that was good news for that bay, but not necessarily a huge gain overall. Looking at the bay as a whole. And this is a little map I put together that just shows, again, looking at the transect data by individual segments, what was the change in frequency occurrence from 2024 to last year, just to show you where some of those gains have occurred spatially by transect. And so clearly the larger green points is where we saw an increase in frequency occurrence at that transect, and red would be a decrease. So overall, if you kind of squint, you can see that the green ones sort of are more present than the red ones. You might notice that, just to point out, in Lower Tampa Bay, there's only 2 transects here, and that's because these transects down here, all but these 2, I think, are monitored every other year, as the I think it's 90 county that does those. So you're looking at Lower Tampa Bay long term. Just keep that in mind. There is sort of this staggered step between years where transects are monitored and not monitored. Although these 2, I think, are monitored every year. So there are those down there that are consistent.
Boca Ciega Bay is also every other year.
Yes. Okay, so that's water quality, seagrass. I'm not going to belabor this point because Carrie did a good job talking about it, but for years we know that the exceedances of chlorophyll in Old Tampa Bay were in part related to pyrogenium blooms. We've seen a reduction in chlorophyll in Old Tampa Bay. That's again related to the reduction in pyrogenium, but not that being the sole driver of chlorophyll. But just to point out, yes, we have seen a decline in pyrogenium. I think it was, 1 point in 2025 where it was observed, but that was it for 2025. So that's not to say we should stop thinking about pyrogenium. It very well, could come back with more, cysts hitting the sediment if that, conditions are favorable for that. But the good news is that, that doesn't seem to be a large driver, at least in the last year or two, for water quality in Old Tampa Bay. I haven't reported on this one yet. I think the last couple of years I have gave hints that this is forthcoming, but we do actually now have a fecal indicator bacteria report card. Last couple of years we've been developing more indicators where we know we have data, but not necessarily a process for synthesizing that into some sort of meaningful communication product for you guys. And so this is the first year I'm actually going to report on this particular report card. Really, it started as a project where there were some methods that were historically available for the Hillsborough and Alafaya basins as part of BMAP for the fecal impairments there. We kind of just extrapolated that method to Tampa Bay as a whole, just to get an overall sense of the exposure risk associated with fecal contaminants. And it does come with some caveats, which I'll explain in a second, but it does grade each base segment into these 3-letter grades, where basically the better the grade, the lower the likelihood of exposure risk relative to a Department of Health exposure standard for Enterococcus. So this is the long-term story. Let me just step back. Just because Old Tampa Bay is all red doesn't mean it's riddled with poop. What this is showing is each base segment is going to be expressed as a function of these long-term monitoring stations that were picked specifically as outfalls to that base segment, just to get a sense of, how much potential Enterococcus may be entering at those locations, not necessarily a description of the, right in the middle of Old Tampa Bay, for example. And we do this because we want, we are going to roll up all of our indicator products into a bay-wide report card. And so we need, to sort of communicate this at these broad spatial scales. We'll think about managing the bay as a whole. So, but really, long-term Old Tampa Bay, Hillsborough Bay, that's where a lot of the urban influences, and so that's where you expect a lot of the fecal contamination to occur. And this is a typical, spatial pattern we see for a lot of our report cards up in the bay, where we have a lot of our freshwater inputs and major inflows in the bay. That's where we see a lot of those contaminants coming in. So just more or less just making you aware that this product is available. There's not necessarily any kind of actionable outcomes yet from this, but it is something you can look at to get a sense of long-term trends and fecal contaminants, looking at the major outfalls to each bay. There is a report card or a dashboard online as well where you can look at the individual stations. So take home, good news, water quality the last couple of years been pretty good, but again, that I think is just a product of lower than average rainfall. So we can't always depend on that, as I've said in years past. That is not a management strategy. If we have a year where we have higher than average rainfall, we're likely to exceed, it's very possible to exceed these targets. So the other thing too is that I did kind of briefly mention this issue between chlorophyll in Old Tampa Bay and light attenuation. There does seem to be a mismatch in what those trends have told us, at least last year. So it could be other things driving water clarity in Old Tampa Bay that we might look at. But yeah, that's pretty much it for water quality, and I will hand it over to Eric now, who will wrap up the rest of this.
All right. Does anybody need to stand up? Take a little break. All right. So I'm going to go over the last couple of report cards that we put together, and a lot of these are focused on wildlife and habitats within the bay, not necessarily water quality. The first two report cards, the Necton report card and the benthic report card, you'll notice that we have data from only until 2024 because there's more processing time that's associated with those two report cards. So just keep that in mind. This is matching up directly with the water quality that Marcus just talked about. So the Necton Index was developed in collaboration with FWC and the Fisheries Independent Monitoring Program. So it takes the information that they take on small-bodied fish and macroinvertebrates and compares it to the average of basically a species diversity and richness parameter, so that we can look at how the Necton is doing in a particular year versus how it has done in the past several. And a lot of times what we see with this index is that it responds to large scale perturbations. So for example, if you look at 2025 up here, that first line that has all three in the yellow, that was a large red tide year. So there was a big difference in the Necton index. So again, those color those colors are associated with action. So green, we're going to just keep what we're doing. Yellow, we need to do more research into maybe why these trends are occurring. We haven't actually had a red year for Nectin, which is good. So that would mean, we need to seriously look at some management recommendations. Again, if you look at, kind of going down the time series a little more to like 2021, you see some more yellow there in the index, and that's also associated with another larger red tide that happened within the bay. So you can see that there's still the Nectin index is still in the yellow right now. Could still be some recovery from that event. We were also interested. So, last year we didn't have the information from 2024. This year we do. We were wondering if there was going to be an impact from the hurricanes. We did a lot of background data analysis, kind of looking into individual, variables that might have affected the Nectin index. And what we found is that there wasn't a huge impact from the hurricanes, at least for this diversity and richness index. A lot of the lower values that influenced 2024 actually came from the wintertime. And the winter is usually when you lower diversity and richness in those samples anyway. And there was also several samples that, were in areas with no seagrass. So anyway, we were hoping that we would be able to have like a more comprehensive narrative to explain what's happening, but just suffice it to say that we're still kind of recovering. We're still in the yellow here, and actually in Lower Tampa Bay we were in the green, which is good. You can actually dive deeper into this if you want to go onto the dashboard, and you can look at individual samples and see what they look like. So you can see, there's more red dots or on alert dots up in the upper bay segments, kind of the same idea. Some of the other report cards, you're seeing more freshwater inflow in those areas, more impact. So if you want to talk with me after, we can talk about like all the different ways we kind of parse out the data to look at this, but basically, that's where we're standing with the Nectin right now. The benthic results, this is an index that reports on the health of aquatic organisms near the bay bottom. So this is an index that looks at all the different animals that live in the sediments, and this index is really sensitive to pollutants that can accumulate in the sediment. So you don't see a lot of difference in this index because a lot of those legacy pollutants are still in those bay segments. So you can see similar patterns to previous years.
Gary, can I say one thing about that? So the way that this is reported this year is a little bit different than previous years. We made a slight change to all of the years after I think like 07 and 08 because of reduced sampling. Basically, I think due to funding issues, the number of sites at each bay segment were cut in half around the 08 time period. And so after that point, we started using, I think, a 3-year rolling average for the categories or the scores that make the basis of the categories. So if you look at the report card from last year, it'll look a little bit different from previous years than what we're showing here. But what this basically does is even out some of the effects of the reduced sampling effort by averaging over the years.
And you do see some improvements over the last few years in some of the upper base segments and yellow instead of the red. So, we're going to continue to monitor this. And there's a wealth of information in this benthic data. So, speaking of T-birth projects and things, this is something that could really be a treasure trove if somebody really wanted to dig into this data in a little more detail. Okay, so here is what it looks like spatially. Again, you can dial into individual stations here as well to look at, index. There's also information on the dashboard about different contaminants and a lot of other information that's taken with these samples. Okay, so Marcus talked about a new dashboard, so I'll talk about a new dashboard as well. This one is not quite complete. If anyone remembered last year, we had a hackathon where folks came in and we said, hey, we want a new indicator. pitch us your ideas. We had a full day long thing where folks came in and took different information that's readily available and developed a dashboard. So our winner, this is what they developed. It's an invasive species dashboard. It's about like 98% complete right now, but not ready for prime time. But we wanted to mention it to you because this is something that we're going to be, improving on and reporting on from now on. The data that's used on this is information from the U.S. Geological Survey, so their invasive species database. And it also pulls in data from citizens from iNaturalist. So because of that, most of these data points you'll see are actually on land. This screenshot is actually plants. So the background color, that's based on ecological values calculated by the Florida Landscape Assessment Model. So it takes in a whole bunch of different ecological values and ranks certain areas based on their ecological value for conservation. And each of those individual points are a data point where an invasive species was found, either from one of those two data sets. And then basically they have a ranking system within that where they take the ecological values and then they take the invasive species, and those individual hexagons that you see up there would be priority areas to look at as far as like maybe doing some management for invasives. So this we're hoping will be a nice tool that folks can look for individual species they want to look for as far as invasive management. One other thing that we do want to incorporate that's not there yet, the area within the bay is empty right now because, again, a lot of these data points are ones that would be taken on land. So we're going to try and work with the Fisheries Independent Monitoring Program to get their invasive species, invasive fish and aquatic species into this as well. There's definitely been some records of invasive species in the Hillsborough River recently that we'd like to incorporate into this dashboard. So keep your eyes out. A link and a report card for this will be forthcoming, and we're going to be improving on it throughout the next year. Okay, so now we get to habitat restoration. So every year, all our habitat restoration practitioners report to us on the habitat projects that they did during a given year. In 2025, 66 habitat restoration projects were completed by over 17 partners. And this is the breakdown by habitat. You can see that the top habitat that had the most acreage and the most projects was in upland areas, followed by coastal uplands. And then there were 6 projects each for mangrove forests and living shorelines. So this gives us an idea of the habitat restoration that's happening each year by our partners. We also can rank this over time. So since 2006, we've been reporting this to the EPA in these different habitat designations. So we can actually report on not only the number of projects, but, what's happening in each habitat. So you can see we've had an increase in projects over time. The last 3 years, we've had, similar numbers of projects. And depending on when projects are ending, there may be more in upland areas. There's been a lot of seagrass projects in the last couple of years, oyster bar projects. So this gives us an idea of what's been going on in the bay over each year. And another line of evidence, those restorations are happening, but they might not be mature habitats yet. One way that we assess the different habitats over time and their changes is through our report card here that is based off of mapping. So the subtitle side that looks at tidal flats, seagrasses, and oyster bars, that's based on every 2 years the Southwest Florida Water Management District does their seagrass surveys. And then on the other side, that's based on the land use and cover also by So we have 2 new years on this. So we've got the 2024 data for subtitle, and we've also got the 2023 data for the land use land cover. The way you read this is within our habitat master plan, we have goals for restoration that we want to meet by 2030. If it's red, we haven't met that goal yet. If the arrow is and if it's green, we've met it already. If the arrow is pointing up, that means that it looks like we'll meet it by 2030. So that's a good sign. If it's pointing downward, it doesn't look like we're going to meet it by 2030. So if you look at these in that last line in those mapping efforts, we've actually got a lot more red and a lot more downward facing arrows in the past year. So what we're looking at here, we've got positive outputs for tidal flats, salt barrens, and non-forested freshwater wetlands, and we've got some work to do on a lot of the other habitats. So each year, what we do is we present a habitat report card, a 2-page report card, kind of like the water quality report card. We've been doing that for the past 2 years. And we've been saying, okay, these 3 habitats should be a priority based on either the mapping or the habitat restoration. This year, we decided to do it a little bit differently. So we actually put together a process for the Habitat Restoration Consortium to give input on that. So I'm going to summarize that real quick. So this is going into a different presentation just to show you what we did. So this new process incorporates comments from our habitat restoration practitioners on the ground, and then we bring that to the technical advisory committee, which is you guys. So the basic idea is to whittle down those, 6 or 7 habitats that were in the red to 3 habitats that we think should be a priority for the coming year. If this process is something that you guys like, we'll continue to do this each year. So there'll be more input from the community. So this was the timeline. We presented a draft framework for making this decision in August of last year at the Habitat Restoration Consortium. We then provided a draft habitat report card with all the information I just talked about, whether it's the habitat that was restored in the last year or the mapping results. And then in December, we actually met in person and we talked about these different lines of evidence and, feasible and doable areas and habitats to restore. So right now we're at finalizing the recommendations. We have some for you, but I just wanted to kind of go through the process that we went through to get to that decision. So we had 3 different decision points here. So first we looked at the mapping information. Then we looked at the habitat restoration that was done in a given year. And then we looked at personal observations from folks that were in the room. So if you want more details on this, you can click on that link in the presentation and it goes through like a text explanation, but I'll go through this step by step right now. So first we looked at the mapping information. And like I said, in previous years, it was kind of somewhat obvious what 3 habitats we should focus on just because of the ones that had the red arrows facing down, but this time we had like 6 of those red arrows. So we wanted to get input from the community. So what we figured out is we were going to take, okay, which habitat types have met the targets for the last 2 mapping cycles. So when you take that one and you look at these squares here, tidal flats, salt barrens, and non-forested freshwater wetlands, those all have met the targets and look like they're going to meet them by 2030. So we're not going to prioritize those. The next step, we wanted to look at the actual restoration over time and seeing what's happening in an individual year. The benefit of doing this is that we can actually get information on habitats that aren't easily monitored by mapping. So for example, artificial reefs, hard bottom, living shorelines, tidal tributaries, those ones that are starred up here. We can't really get that information from the mapping, but we can get it from individual habitat restoration projects each year. So we went through and did basically the same process. When you add that restoration in this year, do we meet the target? And if you take the actual like progress over time for each one of those restoration years, will we meet the target? And based off of that, 3 more habitats came out as either meeting or looking like they're going to meet the targets. Hard bottom artificial reefs and mangroves. Those are actually habitats that we need to stay aligned. So we're not necessarily looking to increase them. We just what we have. So that still left us quite a few habitats that were in the red. So we went to the 3rd step, which was actually getting input from the community. So we put together a worksheet that had all the information from those last 2 decisions. So we had whether the 2030 targets were met or trending towards they're going to meet based on mapping, based on restoration, but we added some more information. So we added, okay, are those habitats located in the reservation space, which is like the border on where we think we may lose habitats due to sea level rise. That's within the 5-foot contour. We also asked our partners in the room to fill out, okay, do you have opportunity to restore these particular habitats? And we had QR codes down there for opportunity areas that we mapped out. And restoration potential by habitat type. So they could use that information as well as their own, personal knowledge, whether it was a local priority for them, whether there was funding available, and whether it was actually feasible to do that in the next year. And then we also had a column where they could actually put, okay, we actually have these projects planned for the next year. So this is something we can definitely focus on. So we got all that information from them. We had really good discussions on different habitats that we should prioritize. One of the conversations revolved around seagrasses. That's one that, we haven't met the target, but, on the ground restoration is not as effective as water quality improvements for restoring that habitat. So, at least from a, on the ground restoration standpoint, that's not one that the group felt we should prioritize for this report card. We also talked about salt marshes where, if you restore a salt marsh, it's turning into mangrove. So, that one gets complicated as well. So what we landed on, we have a draft habitat report card, and the 3 priority habitats based on the information and feedback from folks would be oyster bars, coastal wetlands, and forested freshwater wetlands. And basically we just wanted to introduce this concept on how we're doing this and get your feedback and see if this is something that is you think is useful and to the community and to the technical advisor committee. So these are proposed restoration priorities. And again, you can go get additional information from the website. And that is all I have, all Marcus has. So that's a lot of information that we just threw at you about all the different parts of the bay, but we're happy to answer any questions about any part of the presentation. Yeah.
I just have a brief comment about the invasive species mapping. I think it's a great idea to have a map like that, but I'm a little skeptical of using the iNaturalist data. Because as we know, there's invasive species everywhere, and people are reporting things on iNaturalist where they hike. So I feel like even by just looking at the map that you showed, it's really showing where people hike rather than where invasive species.
Yeah, you're absolutely right. And the consultant went through several different data sources, and basically what they landed on is we wanted to use information where we were going to have a consistent a line of data each year. So, there's a lot of invasive species databases out there, or, articles that are only for 1 year, or, so USGS, it has a long time series. iNaturalist has a long time series. And of course, there's going to be QA that we're going to have to do on that. And it's going to be skewed towards where people are walking. That's why we're trying to bring in other information that's, so for example, the fisheries independent monitoring information we're going to bring in, that would be a consistent scientifically collected database. But Margaret can probably talk a little bit more on it.
Yeah, I forget which TAC meeting it was, but last year we had to come in and present this concept, and we did talk about soil. Basically, Fact of the matter is, it's a good source of data. We don't want to throw the baby out with the bathwater pretty much. We did talk about how we can most effectively use that, because you're absolutely right. It's really a reflection of where people are at, not necessarily a more systematic survey of invasive species. So this is an example of one way we thought it would be useful to use that information, given its limitations. Is we could use it as a way for like early warning, like what is the first instance of particular species, because that's still going to be useful even if it's just a place where someone's looking. So that's just one way we were thinking about overcoming that. But you're asking examples, limitations, and we are actively trying to supplement for systematic surveys like so there's no way around it, but it is a good use of information, a good potential source of information that we are aware of.
Yeah, and we're going to have all of this online. We're going to have the individual data points available online. So if there are instances that the community is like, okay, there's no way that species would ever occur here, we're welcome to feedback and improving it over time too. So, I already looked at it and saw one that I wanted to. So,
I know you guys, and Maya always talks about drought is not a management tool. And the Estuary program doesn't officially advocate, but I think it's clear that we need to support drought conditions, at least in the spring, until which time population reduces in the developed area. It's obvious. It's clear pattern that that's the way to says, if you want a solution, consider.
That's if you look at long-term precipitation last 50 years, this whole region is getting wetter. It's very slight, but it is getting wetter. But there is year-to-year variability. I think just the luck of the draw. Last couple of years have been dry. Good for water quality, good for seagrass, but we can't always count. It's the point. So stay vigilant, keep doing what you're doing. These issues are not going to go away. Susceptible to just look at it.
I just wanted to share some information, and this comes from a completely different data set that we're not tapping into, and I don't know if it's something we should consider. I don't know if Manatee and Hillsborough are seeing the same thing, but so in our dock permitting program, over the last few years, we've started to see a trend of increasing hard bottom and what they call urban corals. We have a delegation from the Army Corps called SAJ 96. And so what basically we're doing is, under certain conditions, we are doing the permitting for Army Corps on like single family residential docks, things like that. And we have to fill out a form, and we have to map out where we're finding these things. And the marine contractor community is very frustrated with it because we're finding it everywhere. Where you wouldn't expect it. So I just wanted to share that. I'm not sure if Hillsborough and Manatee they're doing the similar permitting. Their staff have to go out and do these type of assessments if they're finding the same thing, but we're not using this information, and yet we have partners who are collecting it. It can be it can be in GIS because we're going to specific locations and mapped things like that. So I just wanted to put that out there because something is changing. I don't know what's driving it, no clue, but it came up because we were renewing our SAJ 96, and all of a sudden now we're having to monitor for very specific hard bottom coral.
Be making that data source available online?
I know our team has, because we're required to track it for the Corps, and I don't think so, because again, this is like very specific for dock permitting, but there's no reason why that information can't be available. We're collecting it. We have to. And I don't know if our partner agencies are doing the same thing.
That was for single family docks and what other projects?
Multi-use structures, those types of things. Anywhere where the board would have to issue a biological.
And one thing we're seeing in that process, because we don't have regulatory authority, we do go out and check out these sites with the regulatory people. Even in the Fingerfields and Boca Ciega Bay and all, the contractor for the applicants looking for horses. So they'll say, oh, there's SAB here or something like that. But then we go out there and there are all these corals. And so we're trying to impress upon them, rather than go out and have to redo it or something, if they need to kind of, look for corals along with everything else. It's a resource inventory, not just an SAB inventory.
Yeah, it's a new thing. It's a relatively new thing that's come up. And like I said, we've met with the contractor, we've shown them the checklist that we are required to utilize in order to perform that review for or need for. And it's another, initially when we were going through the renewal process, the Corps wanted us to require relocation. And you have to utilize someone who is certified to do that. And guess what? In the Tampa Bay area, we have literally 0. So they're in Miami. They're all in South Florida. So you're talking about tens of thousands of dollars. And so we had worked with the Corps to find other ways to protect coral construction or relocation of it within the same area, being able to do it, or internally, basically same depth, same water, same light conditions, that type of thing, or that a lot of it is attached to something and moving that thing within that same area and then keeping an eye on it. So it's just very interesting.
I'm with the Corps of Engineers, so I can relate to what Kelly's talking about, although I don't deal with SHA 96 directly, just occasionally do independent projects. But the reason that we are concerned and this is coming up is because hard bottom habitat is a trust resource that's regulated under Essential Fish Habitat and the Magnuson-Stevenson Act. And those are laws that the federal government has to comply with. So, but like Kelly alluded to, seems like kind of a new issue for us. And as an office in Tampa, we've had conversations like, are these corals, these urban corals, new, or are we just now looking for them and are they being reported on? So we're having those discussions. So we're trying to get better science and better background information on that. Because we're seeing corals reported all the way up to Pasco County, which is we have not seen before. And when we have projects come in, now it's like, okay, you not only have seagrasses, but you have these corals, and what do we do besides avoidance, which isn't always possible? Like Kelly said, we've got to look into how we address these and how we mitigate. So the federal government's not necessarily going to be your best source of providing documentation on where these are at through our various projects. That's probably better done at the county level, but we're definitely interested, Kelly, and I know you're having conversations before already.
Yeah, this sounds really interesting. And if you think that this would be something that would be of use to the community for us to try to find a way to track, I'd be interested in talking about that too.
Well, we should move on to the final. In the spirit of making sure that TAC is a vibrant, active committee, we are soliciting feedback from y'all at this point for T-Bird project ideas. So we're going to be doing a lightning round of presentations. That's
Okay, everybody. We're gonna break up into groups. So I'm just gonna have you count off in 1, 2, 3. So
2, 3.
You're not looking, but you're one.
Oh, yeah.
Yeah.
Two.
Back here.
What?
Three. One. Two. Three.
One.
Two.
3, 1,
Two.
3.
One.
Yeah.
2, 3.
One.
Oh, counting.
You're two. You're number two.
Three.
You're three. Just remember. Okay. It's 1, 2, 3. 1. Yep. 2.
3, 1.
All right. The number ones. Come over here by Kelly. Or actually, come over here. Number one. Come over here.
Yeah.
Number one's right here. Number two's right here. Number three's, Blake, raise your hand. Yep, Randy, great, three's over there.
So absurd.
Oh, twos.
Let's just go.
I'm right here. Okay.
For a home. All right, cool. Yeah. What number are you? Yeah. Two. Oh, two. Okay, cool. Yeah. One. That's for the last one. Okay. What's up? Some structures and.
I don't know, I was chit chatting.
All right, we've got our ones over here, twos over there, threes back there. Okay. All right. So today we're gonna try something a little different. What we did is email a whole group of people who have expressed interest in the Tampa Bay Environmental Restoration Fund, either as an applicant or as a researcher, and asked if there was anybody who has a project idea that they wanted feedback on. We got a lot of people interested actually, so we've limited it to 6 that seemed like the most applicable and the people that actually got back to me on time. So that's what we're doing today. We're doing like a little lightning round of presentations. There's gonna be 6 of them. They're gonna have 4 minutes to tell you about their project proposals. And then you're gonna give them feedback or ask them questions about what they presented. So we're gonna do 3 presentations first. Then the people who presented are gonna have a chance to sit down with each group. So you're gonna have like little conversations amongst each other. You also have a document, there's probably 3 or 4 per group. Feel free to write down anything and everything that comes to mind. This is like a no bad ideas in a brainstorm kind of situation. So just put in any feedback, any questions, whatever you might have up as they're presented. And then if it's something you want to talk to them about or ask about when you're sitting with your small group, feel free to do so. So a quick background on TBRF, if you haven't heard of that term, it's Tampa Bay Environmental Restoration Fund, and I'm not going to say that. I'm just going to say TBRF. It is a competitive grant that we manage at the Tampa Bay Estuary program. The focus is on habitat restoration and water quality improvement projects, but it also funds applied research projects and behavior change campaigns. So the focus is on habitat and water quality, but it does fund some other stuff. Some of these are research heavy today, so keep in mind that for it to be a TBRF project anyway, it has to be applied research. So if you are a resource manager in some agency or in some way, ask, like, as they're presenting, would this be applicable to my work? How would I implement the results from this? And if you don't have a clear answer from their little micro presentation, make sure to tell them that. make sure to give them that feedback when they're in your group. Or ask them, or say like, I think if you change this, it could be applicable in this way. So just be very candid. This is the time to just share, give as much feedback as you can. Because we want to make everybody's proposal that much better. So we have even better projects coming out of it. On that note, our first presentation today is Damon Moore. Let me get my timer out.
Yeah, I'm trying right now.
4 minutes. And you.
That's okay. I'm dancing this time.
I think it's the first one, should be the first one on your page. And if you're ready. And I'm gonna be super serious about this timer, so. All right.
Hey, Jessica, do you have for the folks online?
No.
This should have came.
You should have came. No, just if you do have feedback online that you want to give them, put it in the chat because we have a record of the chat, right? So we can download that and send it to the presenters if you have feedback. Cool. And go.
Hi, I'm Damon. I'm the executive director and founder of a small nonprofit group that's been around just over 3 years now called Oyster River Ecology. We're very much on the applied side of making these kinds of habitat restoration projects. I've had a long, fun career doing these sorts of things. That's one of my projects at Robinson Preserve. But we are planning for a T-birth application for a project in Old Tampa Bay this time. I'm leaving the murky waters of Manatee County. And basically what we'd like to do is, like we've seen on all these charts and stuff today, that oysters thing is like, oh, we need more, we need more. And if you look at like the 2050 plan, it's like we need 300 acres more. And like, we're just not going to get close unless we start kind of changing the ways we're doing this, because it's hard and it costs a lot of money. So one of the things starting this nonprofit that we really set out to do was to address those issues. And the one way that we've done that is through this thing called the Oyster Rag Pot. We're in culch limited systems. This is a very cheap and easy way to get that culch back out there into those systems. If you get them in the right places where food availability is high and predator pressures are low, they work really well. We've looked at all of the fancy models that the estuary programs and everything else is built in there. We have our own very dynamic oyster model. In this case, it's Abby. Abby is holding, and I can spread these around the room, but this is an oyster rag pot that's been in the water for about a year on these projects we've done in the Manatee River. We have just over 22,000 of these things out there now, funded through a T-birth award last year to do a full acre there. We have 2 grants out right now, but by the time we get those, hopefully they will be at 5 acres. But they're pretty simple. It's a cement rag that's coated in cement. There's a wooden spacer inside and a 12-inch stake. We take this thing into the bottom, stick it in there, and then those oysters grow off of the side of that. From these areas where it works really well, to about 12 months until they start to coalesce. We are working on a permit right now in just off of the project that Pinellas County did at Philip Park. Randy, waiting to hear back on an email from you about that.
Yes, and I'm going to respond. I had to talk to my regional people.
This is all. But no, we're serious about doing that. And one of the way that we're doing this is it's literally like half the cost of just spreading rocks out there. We're doing this now basically with like groups of 5 people, where we work at a warehouse. I would say we needed a boat, but last night we were awarded another grant to buy us another boat and another truck and another trailer. Thank you to Sarasota. But basically what this would do was would start a new warehouse up in the Pinellas, to serve Old Tampa Bay, the way we're doing this work in Manatee County, and just staff that completely. I would basically run quality control on it, but hire all the staff to run that whole thing.
1 minute.
1 minute.
That's never happened.
I know. That's why I didn't. Was that? But we've been out with Stacy. That like she has a project that she had done there. What we can kind of gauge from that out there is that where they have put their oyster bags out, and you have a lot of good vertical relief, we have good oyster growth. Where they put loose shell out, we have a lot of migration of those sorts of things. Ours have all survived all of the hurricanes that we had out there. We actually snuck a test plot in out there. We might stick out there to see what he's done there. But Abby and I had done like quite a bit of work over the summer, really looking for predator pressure. That seems to be a major driver, mostly with oyster drills and crown conchs, to find those areas where those things are low enough so that these things will actually grow, not just get eaten year after year. One of the best indicators. Anything. I forgot to put anything specific. It sounded like I was being like, I don't want to hear anything.
It's not.
But it's just, yeah, I know. It's there was no specific feedback. I just want you to lay your praises on me and tell me what. No, it's that's my 15 minutes.
Okay.
That's it.
Yeah. All right, thank you, Damon. Next we have Jasmine from Minority Shark Sciences, and she's gonna give you a presentation as well.
Okay.
So when you're ready.
Okay.
Oh, you're right.
Okay. So as we all know, red tide has been a persistent issue and stressor in the Tampa Bay watershed. There's been a lot of investments in mitigation strategies to lower risk of blooms, and recovery strategies to shorten or weaken blooms. But there's not a lot of how these strategies are working across the ecosystem as a whole. Namely, what I'm interested in is brevetoxin, which comes from Carnea brevis, and how this moves through the trophic system. And so we had done a pilot study with our sharks and found evidence that nearly all of our sharks have this toxin in their bodies, regardless of if there was a recent bloom or not, from sampling them. And so we're really interested in how this toxin is staying in our systems, and if they are actually moving up through trophic levels, indicating that the issue may not end once the bloom ends. And so we're really interested in mitigation and restoration strategies that are addressing red tide and how well they actually are addressing this toxin issue. And we're really interested in this toxin because we have evidence that a similar, like dinoflagellate, has produced a neurotoxin that has caused like spinning diseases in the Keys. I do a lot of work with the small-toothed sawfish, and we had obviously a whole lot going on with that. And so that's why I got interested in this ecotoxicology and algal blooms and how issues beyond the crash of the bloom and issues around oxygen in the water could be persisting long after the bloom is over. So we're proposing a 2-year collaborative effort between Minorities in Shark Sciences and New College of Florida, where we'll be doing brevetoxin assessments across multiple trophic levels. So looking at the toxin in the water, in our plant communities, herbivores, and predators, and pairing that with the Carnea brevis cell count data to provide baseline data that we establish through regular sampling, and then obviously picking that up once the bloom occurs to monitor and track the recovery time of the system and see how long it takes for that toxin to kind of work its way out of the system, if it does at all. And this will include water quality surveys, catch and release surveys, visual surveys, things like that. So we're really interested in evaluating mitigation efforts, not just by decreasing cell counts, but also addressing this concentration issue of toxins and their cumulative effect as it moves through the food web. So we want to make sure that these efforts are actually translating to an overall healthier ecosystem, and we're really being aware of these long-term impacts of these blooms. Thank you. So it'll be myself. I'm obviously specialized in sharks and our predators, and then we have Dr. Samuel Bedgood, who specializes in algae and invertebrates, and Dr. Samantha Lavelle, specializing in bony fish. And so we are really wanting to work together to look at this issue holistically throughout the ecosystem, and identify which animals or trophic levels are most vulnerable. And this could help us support adaptive mitigation efforts that tackle not only the blooms, but also their long-term lasting effects through these toxins. And also working with a college, and we ourselves have interns. We're also hoping to involve the next generation of scientists and create opportunities for community involvement in monitoring efforts. And what I want to know is, would this research be helpful to resource managers? If so, how would you use this? And what information could we collect to make it helpful?
Time is up.
That's it.
Thank you very much. Okay. Then Tom Reese, the Diplomacy Spring Restoration Project. Yeah. Go.
How many of you know where Fred Hall Park is? A handful. Okay, it's on Bayshore Boulevard. It's the only green space on the entire route. And thousands of people go there every day. And on Gasparilla, there's 100,000 people there. I bet you less than 3% know that that's a spring. There's 2 springs there. So in the late mid 1800s, those springs were thought to have healing powers. So people came in there and wanted to bathe in those. So in the 1920s, Thomas Palmer, who was a senator, bought the property and put in a little pool access, like we can see here. It had steps coming down on the left, and you could go in and sit in that water. It was crystal clear. It was a spring. And there's another spring on the other side. And so in the 1920s, it was purchased by another individual, and they made a huge pool out of it. It was only a few feet deep, but it was the size of 2 Olympic pools. And people would go there and swim, or at least get into that water. But by 1959, the water quality was so bad, they closed it due to public health concerns. And then they just put dirt over the whole concrete structure. But the 2 springs flow currently to the bay through pipes. There's no habitat value. This is spring here is 8 feet deep. There's a vent down here. The spring comes up, it comes to the side elevation, goes out to the port, and then straight to the bay. We've already measured the water quality in here. They're elevated in nutrients, both nitrogen and phosphorus. And if we can clean up some of this water before it goes in the bay, that's good for Hillsborough Bay. So we did get a grant from EPC to design a restoration project here. And what we're trying to do is combine these 2 springs, because they've always flowed right next to each other, but never were combined. But we can combine them and create a wetland out of this area. We're going to have some water quality improvements and provide the habitat. So what we're trying to do, we have an EPC grant for 100,000, for 50,000 to design it. This is a concept plan. This is not the final thing, because we're still in the design phase. But all we're trying to do is take the concrete out of half the spring. This is the western half, take the whole eastern half out and create a wetland that's both can have emergent and submergent plants in it. So now that spring water will have a chance to have some contact with the vegetation, get some polishing before it goes out into the bay, hopefully removing some nitrogen and phosphorus. We don't know the cost of this. We're still in design, but before our application, we will have a cost estimate. We have raised $200,000 to do this project. Thank you. And so we think that this meets the goals of the Tampa Bay Estuary program through habitat BH1, which is creating freshwater wetlands. And also it addresses water quality improvements, because we will get some cleansing of this water going before it goes in Tampa Bay. Tampa Bay is on the tipping point. Hillsborough Bay is tipping point of when we have seagrass or not. So if we can clean the water up, anything here, we may keep our seagrass beds to be more persistent. So that's it.
Cool. All right.
Thank you.
So thank you. Speakers kind of already broken up. Damon, you're in 1, Jasmine, you're in 2. Tom, if you want to go back to group 3 back here. So we are gonna do another real quick 5 minutes of just chatting amongst your group with your presenter. Give them any feedback that you have, ask them any questions that you have, any way that you can see that they might be able to improve their project, for example. That's the kind of feedback that we're hoping that you, as the TAC, can provide to your person. Stop it. Okay, so go for it.
For y'all.
Yeah.
It's a lot of people. I have a question on yours. So you asked like how will this be helpful to resource managers? Is your, I guess, just backing up, looking at it through traffic levels, and of course, looking at apex predators, top of the bioaccumulation. Looking at it from a human health aspect, as far as if they're in sharks, it might be redfish or snook or things like that, that people are eating. People do eat sharks, but it's a different aspect here. So is it a human health or more of a resource component to it?
Definitely both. So in talking to people, a lot of us are that's maybe not that right. That's of interest, and then also just from an ecosystem perspective, if our throughout the trophic levels we have just a system, that's shellfish, that's bony fish, that's a lot of things that there's feed and a lot of industries that could be impacted. So I think we're thinking about it pretty broadly in terms of human impacts, but also things like fisheries management from our perspective as well. As understanding going to impact important fisheries as well as sharks and
with that broad, what if down the line, with your particular T-bird brand, you're looking at essentially collecting baseline. We have to start somewhere, right? So you're trying to gather information.
Other than engineering features.
Yeah, I think that the goal would be to find partners that have a vested interest in some aspect of this and tailor our baseline data sort of to them. But also have something, because right now there's nothing about rubber-toxin happening really. It's like a single paper like this. And so we're really interested in that perspective who all would be able to use this and what information would they need in order to do what they would want to provide general baseline, but starting from a specific perspective. Resource management archives.
That base and
I know just stuff. Anyone has any place. Let me know.
So yeah, so with FWC and the HAB group, I know our groups, we have like a whole toxins lab and they do ref toxins on from shellfish to probably fish too. I'm assuming, yeah, I was working in that lab, but there are people that the abilities.
Yeah, I'm facing a lot of.
Where is this kind of a
So would this be setting a new baseline or is this?
In a way, yes.
Just updating what we know now.
Not a single
Toxins and trophics, so. It's like also much for the research done, kind of ballpark, the cost was behind to say lost or something like that.
It's very like, so there's I know that there's some work going on in Sarasota Dolphin Research Program on cetacean. And then there's Wellington Sharks on like what she happened to be able to collect. So it's a lot of opportunities. There's not been any like strategized, let's look at this scales. Let's kind of one-offs. And so I'm interested in sort of connecting.
Speakers, please stand up. Counterclockwise moves to the next group.
Thank you.
Clockwise. Yeah, you know where you sit. Okay. Damon. Come on.
That's a good idea.
Come here.
It's more like. Then we take out the fields. What toxins, what are the toxins in that, but.
Come on, over here. This group.
We don't,
Not just a.
like, not just a, we don't.
All right, you have 4 minutes in this group.
Yeah.
Like, what are
you guys doing? Your works.
Oh, we got.
We have mechanisms for how the toxin.
Not chokehold, I'm just hungry.
Not the right person to ask that question, but I'm assuming there's a baseline that there has to be a threshold, right? Like the gel, like here's a threshold.
No, my vote doesn't count.
Stuff like that. Right. But I don't personally know what those limits are, but there were here today.
We're relying on the latest version of the NOAA Matthew LiDAR. Because we're aiming for subtitle.
Yeah, it's because
we don't have to specifically necessarily be connected to title known. because we're looking for these areas that have those low elevation pressures. So I'm like, where I've done it in the Manatee River, honestly, I'd rather be lower because like the deeper I am, when we do have storm events, I feel like the lower is gonna be better, so.
Yeah, so our plans, gut contents.
Because of what, doing the subtitle, too worried about it, honestly, gonna be found.
And then we do have.
I have a question for you. So for Old Tampa Bay, we were looking at potential for oysters as part of the water quality.
Yeah, so we're checking.
So I know there's probably both all the above, but are you looking at this from a habitat, structural oyster habitat? As that contributes to fisheries and habitat complexity, or from a water quality perspective, or does your system lend itself to one versus the other more so?
It doesn't lend itself to one more or the other. When I write the grants, I do it more towards the structural component, because I'm very much on the applied sort of stuff, and I don't want to get tied into doing a whole lot of water quality monitoring, because there's a lot more people out there that are worth at that. I will take it serious, and I'm good at saying there wasn't oysters, and now there's oysters, and then submit my reimbursement request. And I invite all these people with those giant brains that do that stuff, especially the people that want to start trying to measure denitrification benefits and what's going on in the soil and the rich bacterial activity. I'm not that guy. I'm exactly answer your question.
Oh, it does.
Yeah, I like fish and water quality too.
So did you monitored your 22,000 of these impediments? And there's the but
It turned out.
That was like a really scary thing after all the hurricanes. There was an interesting thing we did find with that too, that was because like physically everything was still in flux. But I was like really nervous because like after what was the first one we had that was like the big thing? Allen or something. Yeah, that one. Went out in between hurricanes, it was like a pond. So it was like it stayed fresh long enough that the most recent spat shows all of those had died. So I went out there and saw like some thumbnail size oysters dead. They were on there so thick, I thought it's cooked. When they finally did flake off, the larger ones from the original spats, it would be about an inch and a half long. They were still fine. So like, and I went back to fall in December thinking, okay, I'm going to see bear cement again. I was coated with inch and a half 2-inch oysters, and those are the ones that
the management.
Imagine just starches or what they can handle that pressure longer. They can handle staying close.
Figure out what all right, time is up. Speaker, please shift to the next group. Okay, right over here. Tom, you're out of the hot seat. Head over to that one.
Are y'all the next group? Man, y'all are intimidating. Do you want me to sit on the floor?
Yeah. 1 minute.
Hey, well, thank you. When I know you have many suggestions on how we can make it even more effective.
That's Melinda. Yeah.
Specifically, and any habitat knowledge. She's familiar with this. Actually, it's gonna be a wetland. Now.
So that's why
Is there any
features
that you're? Well, I don't have no so. But why are you putting oysters at the? Yeah.
So the existing flow under. Just gonna.
Downstream, typically.
The turbine. The flow.
So right now,
You're looking like that is the ultimate outfall to the bay.
fairly
Alcohol itself will stay as a change in volume. But oysters there. To get some cleansing. So we're not touching anything.
When you see like the permit application.
But now the water is going to this polishing natural wet.
It's not necessarily.
When the water gets high enough, then it goes out like it has.
Areas in the management.
There's a pretty good flow in there.
The big.
You're not gonna have a lot of
But
residents.
historical model for that.
We're making it so it's such a long is that, and we're putting the second spring in that new. So it don't come right out where all the vegetation. So it's gotta be aesthetically, if not.
You mentioned one of those two springs essentially combined. Did they have the outflow historically? Or is this going to be a new recommendation?
No, it's the same outflow that's always used for the social living, but it focuses on neighbors. But my mind is, I didn't have that water, but there's a spring more close to water. 400 feet to the west, in the stormwater situation. Picking up capacity, and stormwater focus on quality. This is small little lines of pipe all going it over here. Going around, make it so spring, and then they come together and go under the left. So we're going to the road to combine both sides.
Yes, so that's why we have questions. And then.
A storm.
Yeah.
We have that like. Also.
And that's a great question. Literally when we did that spring.
These are kind of sort of
title search.
work. But really just in the one that.
Yeah, small water, upper basin, the one that run separated, but within a few hours of flow of those springs, flushed out, we didn't see any impacts in any of the plants here, because it's just not there long enough.
There's the whole goal.
So I would have that. Another piece we're gonna do is we are, this part is free, make sure it doesn't flood like we see all the time. And it's just kind of higher part, but there is a spring that.
What do we do? We decided to head here
And it's higher than everything else.
Valley, we're gonna go
And
and
well,
day.
parallel to
We always do that.
So now, I was, we can't get into that base. And if it does.
Hold the same vertical surface radar.
Time is up. We're gonna move on to the fourth speaker now. So the previous speakers, y'all can stay where you are if you want to, and then I believe Ananda is next. Walnut is then after that, it's not. Josh. Okay. Oh, yeah, you're next. Sorry. I've only seen you online, so it's okay. Okay, so quickly before we get to the next speaker, I want to ask a question and be super duper candid. You're not gonna hurt my feelings. Is this something that you're finding interesting or helpful? Yes, if so, raise your hand. Okay. Okay, well, I'm gonna send this, but when he sends out the email to follow up, if you have feedback on process changes or different questions to ask or different ways to do it, the engineers. Okay, so on that note. Cool. Go for it.
So, what I wanted to talk about was, basically the keywords in the first, the title slide. That's urban stormwater runoff, which gets nutrients, excess nutrients. And then the next word, which is an important word, is floating treatment wetlands. They are nature-based nature-based treatment systems which help in reducing nutrient. And the third word, an important one, is biochar. So biochar is helping to supercharge the floating treatment wetlands, where plants and microbes come together and try to remove nutrients. With biochar, it's improving the moisture content and improving how the water flows through it, while nutrients are getting reduced. So we have a project, it's the Gulf of America project funded by EPA. We are working with Hillsborough County. We deployed some floating treatment wetlands. Usually This is what they look like. We have a mat and then we place the plants in. And they help in removing nutrients from the stormwater runoff ponds. But historically, people just do it. We are doing it in a more systematic way where we have a control system, where we have microcosm studies, where we are trying to understand what is actually the nutrient dynamics, how much nutrient is actually getting removed with plants and microbes working together. So basically, through these mesocosm studies, we're trying to see how biochar is helping in removing and how it improves the microbial diversity, and how does plant species actually affect the performance, and how these nutrient loading limits and the hydraulic retention time affects the performance. And the future work, we are trying to scale up this whole process and how much maintenance is required, because if deployment is very easy, it's low cost, but we are still not convinced about how much labor cost is required. And then what would potential impacts in the Tampa Bay area? With that, I'll let Kyle speak for the next one.
I'm Kyle. I'm a postdoc in the same department as Ananda at USF. And I'm talking about a somewhat related but different project. So freshwater wetlands are great at removing nutrients from water before they make it to the bay and cause algal blooms. And part of that effectiveness comes from plant uptake. And in these eutrophic wetlands, often it's typha, as you can see in the picture at Aaron's Pond, that dominate. They're highly productive and they do a good job sequestering nutrients, but over time their detritus accumulates, mineralizes, and can actually become a source of nutrients. So our objective is to take that type of biomass and recycle it out of the wetland and into horticultural products. We're currently working off a $246,000 grant from FDP. We've harvested typha at a treatment wetland in Lakeland, but within the Tampa Bay watershed. And our next harvest site will be at Aaron's Pond. But with, oh, great, okay. And with this typha, we've processed it into biochar and compost. We've blended these with perlite to develop a horticultural potting medium that we're hoping can be a substitute for peat moss, which is a product that's very bad for wetlands in the climate. And we are currently doing greenhouse trials of plants grown in this. We're hoping to do further tests. We're looking at the quantity of nutrients that are removed through cattail harvest in our site in Lakeland. We've found that 1 hectare of cattail marsh has the ability to remove 160 kilograms of nitrogen per hectare and 16 kilograms of phosphorus per hectare. And so we're interested to see what's possible in an urban stormwater pond as well. And I have some of the products you can.
Okay. Thank you very much. Josh from Bonnet Springs Park is going to come up next. Welcome.
Thank you.
Take it away.
Got the handouts?
Yeah, okay, pass.
So Bonnet Springs Park is a 168-acre privately funded public park attraction in Lakeland. Sits on the shores of Lake Bonnet and was an abandoned rail yard. And so it's kind of been a story of environmental restoration. And so historically, the city of Lakeland had dumped all their stormwater onto this abandoned property. It flowed into Lake Bonnet and created the dirtiest lake in Lakeland, which is a tributary to the Hillsborough River in Tampa. We built this park and created more runoff issues. We actually collect that stormwater now. We filter it physically. We filter it through heavily planted retention ponds. We created a sustainable irrigation solution by building a 6.5-acre lagoon that captures all that stormwater. We have now learned since Hurricane Milton that the runoff is adding sediment back into our lagoon, slowly filling it in, reducing its efficiency. So we are applying for a grant to create a barrier system and some underground drainage that will prevent the surface level erosion that is washing across our property and subsequently refilling our lagoon, which is a problem for us in the long term as a result of the increased water going into Lake Bonnet. So we naturally fertilize our property, and so when we can withdraw the water, the runoff stormwater from our lagoon, it keeps it from moving downstream. We irrigate with it. That's a good thing. If we lose capacity in our lagoon, then that water will return to Lake Bonnet, where the city was actually granted $43 million by DEP to clean that water source based on the source point of pollution that we corrected. So we are trying to maintain that in the future and prevent the surface erosion that wreaked havoc on us, to be honest, during Hurricane Milton. We don't have a cost yet. Half of this project was completed during original construction. There was maybe a lack of foresight as to the other side of the circulator pathway that would need this curbing in the underground drainage solution. So we are, that's going to be the application for us to prevent future surface erosion.
You got a minute and a half left. Anything else?
Did I miss anything? I was speaking at 100 miles an hour. I didn't know how long it was going to take, so I apologize. And yeah.
Is there any specific feedback?
Yeah, I mean, I think it's if there's, you know, this is I'm not a estuary guy, so you know, any feedback on what this project needs, or you know, the efficacy, is that the right word? What we feel like the value of this is, sure, we would love some feedback on that.
Cost estimates.
We do, yeah. I mean, obviously we're not engineers, so we can draw it out on paper and know what it looks like, but somebody's got to engineer and put a construction cost to it as well.
All right, potentially partnerships as well.
Yeah, absolutely.
All right. Well, thank you very much. And I believe Chris Wilson is not here. Is that right? I think, yeah, he canceled at the last minute. Okay. So we've got time for one more. If there's anybody here that really wants to give a totally unprepared minute project pitch.
I'd come up with something.
You takers? I am too.
Anybody online?
That's not fair. I can't get through that.
Just kidding.
Yeah. Okay, well, if not, then I'll do one. Okay. Okay, so we've got all these different restoration projects, some of them really small scale, like rain gardens, pollinator gardens, whatever, especially for our Bay Mini Grant projects. They get put in, there's a ton of volunteer effort. Within 1 year, they are defective because there's no upkeep, there's no maintenance. So what if we had a team of people that just went around and did maintenance on these kinds of projects? You could maybe call it a swap team or something. What was your
Squad.
squad? If it sounds like an idea ripped off of Damon, it is, because I really liked it when he told me about it a couple of years ago. So if there is anyone interested in doing something like that, I think it could be really fun and really engaging for people to get involved, because they do see this like pop up of really fun project, and then they're like, oh, what was that thing I did 5 years ago? And they drive by and they're like, oh, it's covered in weeds and not doing what it's supposed to do. That's not like a great feeling to have wasted your volunteer time. So I think it could be really fun. That's all I'm gonna say. Okay. Amanda and Kyle.
Since there's
Okay, so y'all both hang out here.
can we split up since there's there were only two?
Yeah, you could. Gosh, where'd you go? Okay, you're already back there. Kyle, you wanna come over here? All right. Give feedback to your speakers.
Sorry, what are either vote 100%?
Well, so we our circulator.
It's all created from the detritus you're collecting.
That's from detritus. That's from the green living. That is an extract this thing and also so.
So
In the greenhouse, heavy tone irrigating table, and it's daily. And so we didn't have anything there to create.
So ideas of seeing, you know, that's really perfect.
And then what's your plan?
Yeah.
What do you want to do with it?
So we're currently doing greenhouse trials, like campus, and oregano. They started 10 days ago, and in 30 days. We made the 5 months difference in the harvest compared to 30 months. So that will be able to say
So,
I'd be able to say how it performs relative to convention.
The proposal is to see effectiveness of how things are broken or
It's a whole systems approach, so it's from nutrient removal
In addition
to
to paving.
the characteristics of the media. And in the case of the biochar, it's meant for sandy soils.
So the idea is to channel all of that.
We have a lot of sandy soils here. So we're also hoping to do some studies in our first project that are looking at how effective it is at limiting nutrient leaching from agriculture.
But the amount of erosion besides are couple
Studying of plant materials. Yeah. Grow up some sand, that's good from across.
And so we.
Right, but I mean, really, there's no shortage of cattail and SEAP, even though the native species is like it's invasive.
Extruded curving is not terribly.
So there's plenty of cattail. And we found that after cutting the green material, especially in the spring or summer, within 2 weeks, it would be free. So bounces right.
Right, and
Like
that's
as long as it's in nutrient
so,
on the water.
we managed to.
As well.
Or like maybe it's all increase the sustainable.
So it's something that the.
Don't right now.
But it's easier to send you from.
Sorry. There, it's something from Home Depot that might help.
It's interesting.
Environment and
One left in your group.
Where how we're growing our seagrass.
Yes, so this right now, it's actually.
Okay, yeah.
Actually
I
inside.
don't currently,
But the hands one is
Have,
part of the
about how the
in the PVC, so.
nutrient back into the environment.
Okay, that's
Now.
another thing we want to look at is cation exchange. These materials
Cleaning it out and then.
and to do a study in the greenhouse to see like if we pour a fertilizer solution through conventional potting mediums through this, does this do a better job at limiting that is free.
we do free concerts, we do
But we also wanted to.
I'm definitely gonna talk to somebody about
the food, want to eat
that
part
sort
of
of
we do
specialized.
STEM education every single.
I've never.
Like it's a whole premise behind this
So.
$100+ million part. Just to make
Part-time.
board in other places. So see over 8 years. This years.
Thank you. Really sandy. Feel free to pass it.
The next one. Yeah, talking about yes, I was like adjusting tonight and everything, the whole.
Oh, I need to come
like, all right, it's time to start it.
And
That's going in a very.
that's made out of both of those products.
Yeah, it's the same.
That we're using is daily.
Maybe last.
Sorry.
At some point, it's a saturation. Not an estuary.
Well, I'm a bean bar, I'm a retired bean bar guy, so.
It is.
I don't wanna.
You have percentages.
So, but I just wanted to, I'm looking for, feedback, because, probably I'm not gonna submit a.
So it came out of the wet here.
Well, so what happened?
He's gonna,
We tried to get the break down.
but it was a last minute decision.
And so we think that in a greenhouse environment, it will probably.
Finding
landscapes and all that stuff, create a whole moving water. And so.
That's why it's
Water elevation.
I feel always grateful for.
I came in and I missed the first round, so then I felt like it was a fair judge, but based on all of them. Poorly timed.
Some of these cases, the
These flow sheets, these would be in stormwater ponds?
Yes, we already have one, stormwater ponds. as I was mentioning, was just we have a Gulf of America project. Serena is the so we have multiple parts in that project. Really,
two peninsulas
in our system, what they really do tell us how they want to utilize from the infrastructure, and we work in an area part of the university.
So that
managed to work
integrated sustainable irrigation solution.
they clean up now we regularly and then we are also looking at different stormwater infiltration units where we can as well as which can be deployed easily. Yes, we are also not yet but we are seeing that it's a completely way. I didn't have time to do the key graphs here on the right hand side where source of
1 minute left in your group.
ammonia we have in stormwater in 7 days.
That's just like a short-term solution.
Study as well along with the actual trying to figure out a way with design
So moving down river, it doesn't have to increase capacity.
What about long-term maintenance, like, so you can do it over 7 days, long-term, like.
so the mesopolitan study just wanted to see how much time because there's any time requires for the person to actually and that is and for long term.
Another thing I want to mention is based on.
It requires some maintenance. We have employed it in Harris one for last 1 year. It required regular maintenance and also a plant for
Of the speakers. That's our last feedback of the day. Speakers, please rise and move to your next group. Thank you, Josh.
We have some photographs, but they are also important sometimes. They pick out the fire. Yeah, which we use.
Jessica, are you gonna
Give and take, right? Okay.
Thank you so much.
And Andy, you're back down. Yeah.
Cool. Any questions, feedback from me?
Yes.
I didn't know.
So that's why we have one way for actually in a.
Just the living. Are there any other plans where? That's just the shoreline, the other things planted, forested.
Well, and actually, that the area that we're doing is an unimproved, wild area right now. The problem is we have 80 feet of elevation in our property and the pavement collects a lot of water and it creates a speed as the water goes across property. And so we actually lost over 8,000 square feet of natural material because so much water got through there and we had like a 5 feet ravine. It just washed out the entire area. And so the idea is to prevent that from happening again. As you can see on the map here, this area right here got just completely wiped out during Hurricane Milton. And so preventing that erosion,
how long
moving into our new where it needs to be for our sustainable irrigation solution, and prevents the runoff from entering back into Lake Bonnet. There's kind of a false man-made wetland between our lagoon and Lake Bonnet that was created 50 plus years ago. The city was dumping all their stormwater on the land. So through the infrastructure changes that we've done, that's the idea is to keep what we've done, prevent it from going back to a negative position where the nutrients and the trash and all that stuff are running from the roadway into Lake Bonnet. We keep it on, irrigate with it, we're using that bad water for a good purpose.
Feels like a big nitrogen load reduction project.
Oh, yeah. Yeah,
I mean, we permitted through everything with them. The funding part was done. So, we don't get tax funding for anything currently. And we did not run construction either. So the almost 9 million we spent on infrastructure was all privately based.
Consider the higher expense. That's causing.
Where the plant?
The accelerant flows.
It's much the elevation, so the land was actually covered with arsenic, and we had to remediate the arsenic, so we didn't do anything else, and so as opposed to buying land and moving the arsenics, we actually worked with DEP to encapsulate them. So now we have 2 mountains on our property, and that's what created that 80 foot of elevation change down toward our lagoon. So our roadway is it's like a standard road, 20 feet wide or whatever it is. That's a big issue, but it's 168 acres. This will converge down to that lagoon, which is at the low point of our elevation. So that's like Lake Bonnet have arsenic contamination in site. I don't think there's arsenic contamination in Lake Bonnet, but it's my understanding, and this was part of the DEP grant that we worked with the city on, that the water column, like there's like 2 feet of clear water, like 18 feet of sludge all the way to the bottom. So $43 million. And the city is to remove all of that, which will increase the capacity of that lake as well. 43 million. And DeSantis came after our property 3 years ago. We're still working on that. City working on it. But the idea is once we get that cleaned up, to stop this cycle.
Time is up. Thank you so much for participating. I have a couple of wrap-up questions for you. Obviously, you can keep chatting after the meeting. We're almost there. If you did like this format, what is it that you liked about it? The direct conversation part, the rapidness, tell me what's good about it. Anybody?
It's lightning.
Lightning? It's fast. Yeah.
Mixing a few fast ones in with these longer ones kind of thing.
Gotcha.
Yeah.
Interactive conversations.
Okay, so is it better in smaller groups? Yeah, okay.
Like interactive conversations, if you're like talking all the time, split it up like this, we break those people up a little bit more and it gives like more ideas.
Any other thoughts on the positive side? All right, thoughts on the negative side. What do we need to do differently or better?
Maybe I couldn't hear some of the other, like sometimes there are other ideas that I want to hear as well.
Maybe like sharing,
summarize afterwards.
Yeah.
Or highlight.
Okay. Okay. Other thoughts on improvement? One more. Yeah, one more.
There were
Just a little bit more time when you're in these groups, because you're getting some good feedback and then it seems pretty quick.
So maybe presentation time, same, a little bit more on discussion.
And I would say there were definitely some questions I liked about, so giving instructions as well, make sure they have detail. Yeah.
Okay. Okay, sure. Okay, that's helpful. Okay, yeah.
I would say sometimes the questions were just trying to understand it more and maybe not, there wasn't enough time to, once we finally understood what the project was, and we had a few questions.
Okay.
Then that's where we didn't have enough time to answer the questions they wanted, because we just needed a little bit more background information. So maybe more time in that conversation spot.
Okay.
And like specific questions.
Do you think it would help if we prep speakers better on what information to provide so that it was you didn't have to ask those kinds of detailed questions in the groups?
Or maybe they have a one pager on
Okay. All right.
And then maybe 1 or 2 minutes for clarifying questions in the group, like they do their lightning talk, and then 1 or 2 minutes of any clarifying questions, and then make the small groups more targeted feedback.
Okay.
So that might help.
Thank you for this experiment. If you are gonna apply for T-Birth, there's an informational webinar on February 4th. We do have a new format this year, hopefully streamlined and even easier than it has been. So come learn about that, and then the application deadline is March 25th. Thank you very much.
Blake has an announcement as well.
Yeah, so you guys might know that soon we're going to be doing our 2027 revision of our CCMP, our Comprehensive Conservation and Management Plan. Over the next year, we're going to be really zeroing in on updating all of our CCMP actions. The first task that we have to help us gather information for starting this request is what we're calling the Baywide Satisfaction Survey. And so that is going out to members of the public as well as to all people within TBEP's network of experts. And so that is going to be opening on February 1st. We already have over 800 people from the public that have shared their thoughts on what they would like to see for the next 10 years. So now we're going to open it up to you guys, people from our Habitat Restoration Consortium, Community Advisory Committee. So February 1st, that'll be coming out in our newsletter as well as on our website. If you want to go take it and share it with folks that you know, you can give us feedback on past CCMP actions and priorities for the next 10 years, as well as some other things to help us kind of figure out what our roadmap is going to be for the next 10 years. Please share within your networks. Thank you.
One last thing, if you have the paper, just leave them and I'll pick them up. If you have any last minute thoughts from your speakers, they are anonymous. So, last little feedback.
Anybody else have any other announcements or anybody online for the good of the group? Okay, well, thank you all for coming. Thanks to those online that stuck through it. And Ryan.
Meeting is adjourned.
Next meeting is March 18th.
Yeah, it's black.